Okay, we're going to get started in a few moments. Encourage everyone to have a seat. And good afternoon. For those of you who are perhaps just joining us, I want to welcome you to the 2020 Breakthrough Prize Symposium. This is a fabulous event, and I'm going to be moderating for the next afternoon session. My name is Tejal Desai, and I am professor and chair of bioengineering at UCSF. And as a bioengineer, this is especially exciting to me as someone who has really been interested in bringing together physical sciences with biological sciences. We're seeing the whole gamut of that today, and really seeing how fundamental advances in all these different disciplines are really going to change the way we interact and perceive of the world. I'm especially excited to have this afternoon session, which again showcases how fundamental science has really been transformed by interesting observations, which maybe otherwise would have been overlooked had it not been for our wonderful scientists here today. So I'm going to begin the session by introducing Daniel Freedman. Daniel Freedman is an emeritus professor of physics and applied mathematics from the Massachusetts Institute of Technology. He's also a visiting professor at Stanford University. And he's one of the winners of this year's special Breakthrough Prize in fundamental physics. And this is actually an occasional prize. It is only offered or awarded for extraordinary achievements in physics, but can be awarded at any time. And so now is the time. And this year's special prize recognizes the 1976 discovery of supergravity, a theory of fundamental particles and forces that has really proved highly influential and has shaped generations of scientists and physicists to come. The title of his talk is, Does Nature Know About Supersymmetry and Supergravity? Why don't we give a round of applause?
Thank you very much. Well, the title's been modified a bit, as you can see. Well, when you're 80 years old, you have a lot to look backward to. And I say that to indicate that I might prefer to talk about the past, but I'll do my best to address the future. What I plan is a qualitative argument for the realization of supersymmetry and supergravity. It's not a rigorous argument. It wouldn't satisfy Alex. But I hope you find it's a vigorous argument, and it's one that I've really felt compelling through the years. But I have to caution you, because until a few years ago, many theorists found the vigorous argument called naturalness very compelling. This argument predicted that at least some of the heavy particles predicted by supersymmetry and required by supersymmetry would be produced at the LHC. And that did not happen. So theorists can talk, but nature speaks last. Let me begin with a few definitions. These are very informal definitions, and I think it'll help to set the tone. I want to talk about two symmetries, global supersymmetry and gauge symmetry. In a global supersymmetry, the transformation you make, the parameters of that transformation, which could be an angle, are constant in space-time. And in a gauge symmetry, they are arbitrary functions of space-time. So you can think of a checkerboard, and on that checkerboard, you have a bunch of needles on every square. And a global transformation would be you rotate every single one of those 64 needles by 30 degrees. Gauge transformation, each square can undergo a transformation by whatever angle you dial. Now, global symmetries are important, but gauge symmetries are more important because they have historically and mathematically led to new forces in nature. So global supersymmetry is the only symmetry principle that that relates particles of different spin. I'm sorry, I can't focus very well on that. It is a candidate for the physics beyond the standard model, but it is not yet confirmed because the heavy particles have not yet been detected. Supergravity is a gauge theory, which combines supersymmetry and general relativity, and it brings gravity and quantum physics closer together, quantum mechanics closer together. So here's my argument. Gauge principles play a vital role in quantum field theory. Quantum field theory is a language we use for particle physics uniformly. There are rigorous theorems that tell us there are only 3 gauge principles that allow interactions among particles and with gravity. There's a spin 1 gauge principle, which is realized in quantum theories of electromagnetism and the weak and strong interactions. There's a spin 2 gauge principle, which is very important, indeed vital to the classical theory of gravity, which is Einstein's general relativity. And the only one permitted by theorems is the spin 3 halves gauge field, and it's that gauge principle that's embedded in supersymmetry and supergravity. So SUSY, as I more or less said, is a very powerful symmetry that relates particles of different spin. Every known particle has a heavier superpartner. Negative LHC results on the searches for those particles have come up short, and that means that the superparticle masses must exceed 2 TeV, which is the energy range of the LHC accelerator in Geneva. And just for perspective, 2 TeV is 16 times the mass of the Higgs particle. Whoops, I think I went too far. I'm sorry, I skipped too soon. Let me just say that an elementary particle is described by a quantum field. It can be massless or has to have a positive mass, and quantum mechanics forces the spin to be either integer or half integer. There are 2 classes of particles in nature. The bosons all have integer spin. They include the Higgs, newly discovered a few years ago, which has spin 0, described by a scalar function in space and time. The photon has spin 1, it's described by a vector field. The graviton has spin 2, and it's described by a tensor field, a field with 2 indices. Fermions are also abundant if they have spin 1/2. Among them are the electron, the quark, they all have spin 1/2. It's also described by a different type of 4-component object, the spinor field psi alpha of x. If the gravitino is eventually discovered, it will have spin 3/2, and it will be described by a vector spinor, 4 times 4 or 16 components. Let's try to define the spin 1 gauge field, if I can focus on it. The photon is described by a vector field, a mu of x, and you make a transformation in the equations of electromagnetism. Everywhere you see a mu, you replace it by a mu plus the derivative or the gradient of theta, where theta is the gauge parameter. It's an arbitrary function. And the equations of electromagnetism remain unchanged under this transformation, and that invariance properties of the equations ensures that the interactions are consistent. The spin 1 gauge principle, we've already said, governs the 3 forces which are explored by accelerator physics and at the LHC accelerator. And that experimental exploration and the accompanying theory have led us to the standard model of those 3 forces. I should emphasize that the electroweak and strong theories are quantum theories. I will not attempt to define quantum theories here. I'll just give the buzzwords, probabilities, particles as waves. In the 21st century, you must use the word entanglement. General relativity is a classical theory, on the other hand. You can in principle, make measurements to arbitrary accuracy. Its gauge field is the metric tensor, which measures distance in space-time, and it has a related gauge transformation. They always involve derivatives of a gauge parameter, but the metric tensor changes by a symmetric derivative of a gauge function xi mu of x, which is an arbitrary vector. Measurements of the bending of light by a star show that the graviton has spin 2. General relativity is a beautiful theory. It's widely applicable. Some of the applications are listed here. As time goes by and tests get sharper and sharper, Einstein's theory looks better and better. However, gravity must be quantized. One cannot accept a world in which 3 of the fundamental forces are quantum mechanical, hence probabilistic, but while gravity remains classical and deterministic. Among the many difficulties associated with quantization of the gravitational field are uncontrolled infinities in quantities that are in principle observable. Okay, now comes the star of the show, supersymmetry and supergravity. Global supersymmetry is a theory in flat space-time with gravity ignored. And the force between 2 elementary particles is so weak that it's a completely safe assumption in practice to neglect gravity. In principle, you cannot neglect gravity. Gravity must be quantized because otherwise you don't derive the force that's holding us to our chairs. Here's an example of a supersymmetric system, a scalar field phi and a spinor field psi. And SUSY transformations mix scalars with spinors. You see the phi changes into psi multiplied by the transformation parameter. It's a global symmetry, so the transformation parameter is a constant 4-component spinor, and psi rotates into a gradient of the scalar field, again times epsilon. As in many symmetries, as in physics, the important structure is revealed by making a repeated transformation. First, you let phi, say, transform with parameter epsilon prime. It turns into psi. You then transform it again with parameter epsilon. It turns back into phi again. You subtract the order of the two, and you find that, as I said, phi goes into phi. Similarly, psi goes into psi. But the space-time point is changed. It's translated by an amount xi mu, where xi mu is a constant vector formed by the spinors epsilon bar and epsilon prime. So what's happening here is that fields are undergoing a translation in space-time. So symmetry thus unifies the quantum mechanical treatment of spin with space-time geometry. So SUSY certainly involves the structure of space-time, and so does gravity. We all know that from Einstein's GR. So this is at least a hint that a marriage between global supersymmetry and general relativity will be a happy marriage. But a constant spinor is not allowed in a gravity theory. It's incompatible with aspects of the spin-2 gauge principle. You must promote epsilon alpha from a constant to an arbitrary function of space-time. So this means that, and this is the ingredient of supergravity theory, supersymmetry must hold as a gauge symmetry. So let's talk a little bit more about supergravity. Not much. I can't show the real equations. They would fill more than one transparency. The supersymmetric partner of the gravitational field is the gravitino field. It's a vector spinor, and its gauge parameter is an arbitrary spinor function of space-time. Here are the gauge transformations. You see that psi is the gauge field of supersymmetry. Its transform involves the covariant derivative of the gauge parameter. So you find the derivative structure common to all gauge transformations, and this quantity omega mu contains derivatives of the gravitational field. And Einstein's metric tensor rotates into something proportional to epsilon and the gravitino. So it is in this way that the graviton is identified with its partner, the gravitino. So this is n equal 1 supergravity with one graviton and one gravitino, as we found it in 1976. So this ends my basic argument. Let me recap it. What I say is that supersymmetry utilizes the the only consistent gauge principle that has not appeared in nature. It unites space-time geometry with quantum mechanical spin. These are very deep principles, and that justifies my hope that experiment will eventually confirm it, perhaps even in the next 10 years. There's one more slide I would like to show to illustrate the further development of the theory. There are many, many different models which have been constructed in which you take the gravity multiplet, spin 2 and spin 3 halves, and couple it to lower spin multiplets, 1/2 0 and 1 1/2. And there are many applications to accelerator experiments and dark matter searches. There are also models of extended supergravity. They all contain one graviton, but they contain any number up to 8 gravitini and lower spins. Okay. The maximum theory and max equal 8 has special properties with respect to its ultraviolet structure. It is proven to be free of ultraviolet divergences up to 6-loop order in Feynman diagrams. And some people believe, perhaps not me, but some people believe that this miracle of a finite theory of gravity holds to all orders. There are also models in all space-time dimensions from d = 2 up to d = 11. That's the maximum space-time dimensions which permit this principle to be realized consistently. And that raises the question of what is the relation between supergravity and superstring theories? Well, let's just say that they've had a very, very productive relation for the last 43 years. There are many, many applications, to black holes, to inflationary cosmologies, many applications to the ADS-CFT correspondence, which has pervaded theoretical physics for 20 years now, and even to new mathematics. And there's one number that says it all. There's the INSPIRE archive, which tabulates physics papers on a daily basis. It lists, over the past 43 years, 15,702 papers in which supergravity enters either in the title or as a keyword. So you see, it's too big to fail. Thank you very much.
Hello. This side, on your right, Dan.
Yes?
I was wondering if you could tell us a little bit more about, you said it had inspired some new mathematics. Can you tell us any more about what sort of branches of mathematics?
I'm sorry, I'm not understanding you.
So you mentioned that supergravity had inspired some new mathematics outside of physics. Could you tell us a little bit more about what kind of mathematics?
New types of manifolds and differential geometry. They come out of n = 2 supergravity. And the mathematicians have embraced them.
And is that something that mathematicians and physicists work together on, or are mathematicians kind of off on their own on that one, leaving the physicists in their way?
I would say both mathematicians and physicists have further developed the issue of special geometry manifolds. I think I see a question right here. I don't see a microphone.
Thank you. So what do you think should we do if we didn't find yet those supersymmetry particles in the LHC? So what do you think does it mean, or what should we do in the future in order to find them, in your opinion?
We can do one of two things. We can build higher energy accelerators. That's an expensive and largely highly political issue. There's also a question of what is the particle responsible for cold dark matter. Supersymmetry models have a candidate, and it's also possible that the gravitino will be cold dark matter. Those experiments take place in deep mines. Their budgets are measured in a few millions of dollars rather than a few billions of dollars. They've been going on, the searches have been going on for about 20 years yet. They haven't yet yielded positive results. but I hope they get lucky.
Hey, Dan, with the, it sounds like the graviton you believe is pretty well established that it's there, and the gravitino is the one that's in question. Is that a fair statement?
That's true. No one should doubt the graviton.
Okay. That should be on your bumper sticker somehow.
Sorry.
If the gravitino is a candidate for cold dark matter, does that mean it doesn't interact with a photon at all? And is that true for gravitons also?
Well, the gravitino is neutral, so it does not directly interact with photons. Thank you very much.
So what a wonderful way to kick off the afternoon. I think this perfectly illustrates how fundamental discovery leads to very unexpected advances in science. And who could have predicted in 1976 the impact of this work across all fields, not just in the physics community, but beyond. So with that, I'm going to switch gears a little bit and introduce our next speaker. Arthur Horwich is the Sterling Professor of Genetics and Professor of Pediatrics and an investigator of the Howard Hughes Medical Institute. He's a molecular biologist who studied the mechanism of chaperone machines that help many proteins in our body fold into the correct three-dimensional space. And it's his co-discovery of these processes with Ulrich Hartl, who will be speaking later on, that was recognized by this year's Breakthrough Prize Committee. More recently, he's actually focused on protein misfolding as a cause in neurodegenerative disease, and we're going to hear some of that work today. The title of his talk is 10-Year Vision, Improving Upon Natural Protein Quality Control in the Setting of Disease. Please join me in welcoming Arthur.
I'd love to have it. Can I keep it? Yeah. I wanted it to be wood so I could really hide behind it. I'm not used to standing over an audience, and I already have stage fright as it is. So I'm hugely honored to be here. And the mandate was to speculate somewhat on where things go from here, and not really talk about my own work. So that is what I'm going to do. And so this talk will have some limits, and you might even object to some of the things that I suggest. So that'll make for interesting questions. So the question that this deals with is, we know something, and I'll summarize in the first few slides. We know something about the quality control machinery in cells that allows recognition and coping with misfolded proteins. But there seems to be situations where that's just not enough, and people wind up with disease as a result of misfolded species that aggregate or have toxic effects on cells. And so the question is, what can we do to go beyond what the natural system is to improve it to help our patients who suffer from a variety of clinical conditions related to these mishaps? And so what I'll do is summarize over the course of these slides, first the natural biology very quickly in a couple slides, and then spend a few slides on what I think we can reach, what has a good possibility of being reached in the next 10 years, what might be reachable maybe over that period of time, and what I see as things that are relatively opaque still to us, where we really need some better understanding if we're really going to be able to go further forward. Okay, so first, I'm dealing with the last step of information transfer here. namely the step of protein folding. Just to quickly reiterate, Ulrich and I made some early discoveries that suggest that protein folding is not necessarily always a spontaneous process, as the early experiments of Anfinsen and others had indicated, that is within cells, that there's a machinery that provides kinetic assistance to protein folding. That is to say, though, that Anfinsen's principles are absolutely correct. I mean, the polypeptide basically can find its thermodynamic minimum in a dilute solvent in vitro, typically. But in cells where conditions are relatively high temperature and high solute content, many proteins really do need assistance. They wind up in kinetic traps, and there's a need for these machines called molecular chaperones. So just in a quick slide, the principle of molecular chaperones revolves around the normal structure of folded proteins and states that are not folded. So a folded protein, typically as shown at the top of this slide, has a greasy hydrophobic core that's buried to the interior of the protein. In this particular hexagonal shaped protein, which is not necessarily a reflection of what polypeptide chains look like when they're folded, but it has a watery surface facing the solvent aspect. And so the idea is that when you have a newly made protein, you're exposing some of these greasy surfaces, and related to entropy and the ordering of water around these unfolded states, proteins tend to aggregate to each other through these hydrophobic surfaces. So the additional production of these kinds of states can occur under stress. So the first things that were studied historically were thermal stress of proteins, causing them to unfold or misfold and now aggregate with each other. So heat shock was the traditional experiment carried out in yeast or E. coli in the early days, as well as mammalian cells. And in addition, importantly to this talk, mutation, altering the amino acid sequence of a protein from its normal state to an altered state that begets misfolding, it simply can't fold in the first place, also begets aggregation via exposure of hydrophobic surfaces. So the principle behind molecular chaperones, not just the ones that we've really focused on, the ring-shaped machines called chaperones, but also a whole set of other chaperones that Ulrich and many others have worked on, is that the chaperone itself proffers a hydrophobic surface. And so that essentially meets the hydrophobic surfaces of an unfolded, misfolded protein, masks them, and prevents aggregation from occurring. And then usually through the offices of energy supply, namely ATP, the chaperones release a protein to try to correctly fold on its own, more or less, or fold, in the case of the chaperonins, inside of a sequestered cavity where there's simply nothing else to aggregate with. Okay, so these are the sort of masters of, or effectors of quality control, if you will. But at a next level, there's a circuitry, and there's been a breakthrough prize conferred on this very recently, that responds to misfolding, for example, in the secretory compartment, ER stress as it's called. Peter Walter and many others, Kazumori and others, worked on this particular problem and dissected it in the simplest forms into really two responses. So on the left, you see that ER stress through a series of sensors, 3 sensors, ATF6, PERC, IRE1, you don't need to remember the names. But the point is, begets a transcriptional response that produces more of this folding machinery that I talked about, more of the chaperones are put into the ER, countering the stress of misfolding. And on the right-hand side, you can see that there's also a second response, which is to decrease the protein load that's coming into an afflicted ER that would beget more misfolded protein. So namely, you're diminishing the level of translation to prevent the amount of substrate that would become misfolded and be a problem in the ER. So there's then 2 levels that we're talking about. One is the circuitry that controls the level of these machineries, and the other is the machines themselves. And so that's the way one counters stress from a variety of standpoints, as well as the standpoint of having a genetically altered protein on board in the cell. So clearly our natural chaperone setup is not able to deal with the problems most visibly to the community at this point in history of neurodegeneration. We're seeing characteristic proteins aggregating within characteristic cell types in the nervous system in a variety of age-related neurodegenerative conditions, as Virginia beautifully articulated this morning from her own work. So for example, Parkinson's disease typically features Lewy bodies in the cytosol of striatal neurons that are composed frequently of a protein called alpha-synuclein. And the Alzheimer peptide, for example, is prominently present in extracellular plaques in Alzheimer's disease as the product of cleavage of the amyloid precursor protein. And I won't go through the rest of these, but it's sort of a characteristic association of a particular protein that goes wrong, can't be properly folded, and aggregates in a specific set of neurons in a specific condition. Okay, so what can we do to improve on protein quality control during this next phase of biology here? So I'll talk about first a series of 4 or 5 things that I think are within reach, then a couple possibilities, and then I'll close with the challenges. So I do think that during the next 10 years, we will see modifications occur at the DNA level in somatic cells that will be able to correct at least some things, and maybe quite a variety. A Breakthrough Prize got accorded to the CRISPR work. And talking to Jennifer the other night, she seems pretty confident that some good things are going to happen in the next few years. So we could think, for example, about correcting sickle cell disease. So a beta globin gene that's containing Pauling and others identified this back in the late 30s and 40s. A valine 6 to glutamate change would correct sickle back to a normal beta globin molecule. And so a direct repair is a consideration. So one would take patients' hematopoietic stem cells, maybe from the red cell lineage in particular, and literally fix them, correct that base change at the DNA level. And now instead of making a sickle beta globin, you're making a normal beta globin. And so the tetrameric hemoglobin is now not liable to aggregation and misfolding, and sickling would no longer occur. Another way to actually rescue sickle cell disease from the Orkin group and others is to actually induce fetal hemoglobin, the developmental form of hemoglobin that the fetus carries, which is obviously able to carry oxygen, and in high levels is perfectly capable of mediating adult life. And so the idea is you're overexpressing and maintaining the expression of fetal hemoglobin into adult life. And so one can do that by inhibiting, as Stuart and his co-workers have shown, by inhibiting the expression of a repressor called BCL11A. So that's some of their recent work from the past few years. And that seems like almost an easier CRISPR project, because there, even an indel at the level of the DNA would be sufficient to produce nice levels of gamma chains and of a decently carrying oxygen carrying fetal hemoglobin. So I think I should also mention that at the RNA level, there's been terrific progress, again, recognized by a Breakthrough Prize last year, with being able to shut off particular inimical forms of genes at the RNA level using antisense oligonucleotides or RNAi. And in fact, in an area that we've been working, at least in mouse models with, ASOs have actually gone into human phase 3 trials in SOD-linked ALS, and that may turn out to be a quite promising direction. So now, another sort of next level consideration is to use small molecule ligands. And so a dramatic use of a small molecule ligand to stabilize a native state of a protein and prevent it from unfolding, misfolding, and actually going to an amyloid fibril state that you see at the bottom of this slide concerns a protein called transthyretin. So Jeff Kelly and co-workers have, over the years, been studying this protein and have delineated a pathway whereby the normally produced circulating tetramer of TTR becomes disassembled, and the folded monomer misfolds over time, and this is an aging-related condition. Also, mutation can involve this protein, and that leads to earlier onset kinds of conditions. But the idea is the misfolded monomer now assembles into an amyloid structure. And so you can think about this whole long pathway resulting in amyloid as being able to be reversed if you simply stabilize the native tetrameric form by putting a small molecule into that ligand pocket that's shown in the blow-up in front of the first image there. Normally, thyroxine would bind there, but a drug called tafamidis binds quite efficiently and stabilizes that molecule. And so for patients with inherited forms of TTR, it seems like this is improving their clinical outcome in early clinical trials that are being conducted. Tafamidis is clearly appearing to be a relatively safe compound to give to people over long periods of time. So another possibility are PROTACs. These are protease targeting chimeras. So you see the small molecule chemical compound there that's sort of an adapter. That adapter brings what's called an E3 ligase that's capable of directing a protein for proteolytic chewing up at one end. And at the other end, it binds specifically to phosphorylated toxic forms of the tau protein that's involved in a variety of conditions that Virginia referred to this morning. So these kinds of adapter proteins are under drug development on the Yale campus and in a considerable number of pharma laboratories. So basically what they do is they recruit an E3, which has an E2 ligase bound to it, and the protein becomes ubiquitinated through amino groups, typically on lysines, and as a result of that, it's conducted to the proteasome where it's degraded. So this might be an efficient way to remove tau, and I'm told that there are some new adapters of this sort that easily cross the blood-brain barrier, so they could be put into trials fairly soon. Another remarkable experiment that actually you'll notice we commented on recently came from the Broad group. Devella Levitt, Greca, and co-workers published this in Cell a few months ago. A remarkable experiment where they said, let's just make the misfolded species go away. We don't want to see it in cells. We have cells in culture, and we're just going to put a small molecule library on the cells and look for ones that no longer have the misfolded species present in the cell. And lo and behold, they came up with a compound that essentially, in the top panel, you'll see has a stalled version of this mucin protein in the early portion of or in the ER-Golgi interface, basically. It's stalled there, and it accretes in cells. You can see it with antibodies as a heavily occupying and toxicity-causing state that leads to kidney failure. They could make it go completely away, and we're able to elucidate that the way this works is by their drug, which is that little green object in the bottom panel, binding to the cytosolic tail of a cargo receptor, competing against what are called COP1 coats, and essentially enabling the protein to just leave the entire secretory pathway, go to lysosomes, and be completely degraded. So that's probably headed to a clinical trial. Really a piece of wonderful work. And then our moderator for this morning, Peter Walter and his group, have also, at the level of circuitry, identified a remarkable molecule that is able to essentially restore translation under conditions of stress. So the ISRIB molecule you see there is a sort of a two-headed molecule, a symmetric molecule, is able to bring these early forms of EIF2B together into what is an active complex. I think it's either an 8 or a 16 mer. Peter can correct me as we go. But that ultimately matures in the presence of the EIF2B alpha 2 molecule into an active EIF2B that allows translation to proceed. And so you go ahead and make proteins under stress, and that actually, under a variety of conditions, may turn out to be a very salutary effect. So that's really a remarkable molecule coming from his group recently. Okay, so now a couple possibilities that are at a really much earlier stage, but can't be completely ruled out. One concerns an actual molecular chaperone, one of our friends, called HSP110, that has been shown to be able to cooperate with what's called a J protein and an HSP70 protein. That's sort of a monomeric hydrophobic binding chaperone. And in the presence of all three of them, you seem to be able to set up a cycle that can actually mediate some disaggregation, maybe even of fibrillar aggregates, as this particular paper from the Bukow lab in the year 2015 shows. So it seems that this is a dissociating agent that's able to actually break apart early, at least early aggregates and maybe even later ones. It seems like HSP110 is the limiting factor, and in an experiment from our group a few years ago, we overproduced HSP110 in an ALS misfolding mouse model, and it increased the survival of those mice by at least a few months and on up to roughly doubling their survival. And more recently, those same mice have migrated into our neighbor Sriganga Chandra's lab, where there have also been effects in vivo in alpha-synuclein mice. So maybe this has some possibility. The problem is, how do you overexpress a chaperone that's roughly 100 kD in size? That's a big protein. It's not clear how to really do that in the nervous system and how to deliver somatically and get efficient expression. Another remarkable experiment that I think renders at least some possibilities related to neurodegeneration is an experiment from Steve McKnight's group that's been carried out over a few years, where they simply asked, let's find a compound that is pro-neurogenic in the hippocampal dentate gyrus. So on the left, you see, at the very bottom of that slide, on the left, you see that there's not a whole lot of BUDR-related mitotic activity going on in the dentate normally. But in the presence of this particular compound, it's drastically enhanced such that you're now getting significant neuronal mitotic events occurring. So that was gained by actually doing an in vivo screen, where they took pools of 10 compounds at a time and delivered those with a catheter into the central nervous system of mice. So each mouse essentially scored 10 compounds at a time, and they scored roughly 1,000 compounds in that experiment, which was conducted a fair while ago. I would submit that high throughput experiments of that sort could probably be done way more efficiently now, and could be used to target mouse models of a variety of aggregation conditions, almost as a black box approach to just trying to rescue something and then deconvolute what the chemistry is doing. Okay, so I'll finish with just a couple of comments on what on this last slide of things that I see as being major challenges, particularly in the area of neurodegenerative disease. I think we don't really have a very good idea of what is dictating region and cell susceptibility at this point in history. For example, why does A beta seem to be so inimical to hippocampal cells and alpha-synuclein to basal ganglia, these as end locations? I mean, Virginia commented on migration this morning, but it's end states where a lot of damage and cell loss is occurring. Why? The nature of spread, as she commented on this morning, is really critical to understand. What is it that Brock was actually describing originally? Is it a prion-like spread, where prions have been shown to move cobblestone-like up the spinal cord into the brain of animals that were exposed to CJD and other kinds of prions? However that is working. Is it a cobblestone type of thing? Is it a circuit-based kind of pathway, as I think some of Virginia's experiments have suggested? And most recently, well, actually, Brock proposed this originally, that Parkinson's disease might be a gut-originated condition, and some recent experiments from the Dawson lab in mice have sort of tended to support that idea that you could at least force things to go that way. The roles of A beta and tau definitely need to be resolved as relates to Alzheimer's disease. Do you need both of those to really progress the disease? Is neuronal activity critical to affecting cells, as was suggested by a science paper back in August looking at short-term delivery of A beta peptide and monitoring which cells were most susceptible? So we really need to understand the nature of tissue and intracellular versus extracellular sites, mechanisms of toxic action of misfolded species in those contexts. Why do you lose synapses? How do you get cell loss? And finally, a really important aspect of all of this is how does aging really play into this? How does it correlate with misfolding and aggregation? And of course, is the quality control machinery itself becoming less competent with aging? I mean, some experiments that Ulrich has carried out a while ago suggest that in model systems like C. elegans, there is a really striking loss of the quality control machinery with aging. So I'll close on that, and the idea is it's somewhat food for thought. We'll see where we go. Thanks very much.
Here? Yeah, okay. That was a very beautiful talk. Thank you very much. I was intrigued by, well, I know a lot about chaperones inside of cells, but I was just thinking of all the aggregates that you talked about, A beta are the only peptides that aggregate outside of cells. And then I realized I knew nothing about extracellular chaperones. I assume they exist, but I don't know for sure if they do exist. So would you clarify that?
Well, there's been some discussion that there are components, apolipoproteins of one sort or another, that might have activity as extracellular chaperones. I think to date, though, we really lack the kind of genetic experiments, which would really probably best be done in a mammalian system, that would really support that in a nice, clean way. So it does seem that there are proteins that can interact with misfolded species outside the cell. But for example, I don't think we have an ApoJ experiment that's really a sort of animal experiment that would really nail this in. But I've got to confess, I could be completely wrong about that.
Is this on? In your last slide, you gave a drug that increased hippocampal dentate gyrus proliferation. But I noted in that slide where there was a lot of BRDU that there was no increase in the thickness of the dentate gyrus. So are neurons dying as they're proliferating?
That's a great observation. I really don't know. I mean, it would be interesting to go back to their published work and see if that's the case. Because I agree that with that level, although I think those are relatively short-term experiments, so that's occurring only over a few days, I believe. But over a longer period of time, it would be interesting to see if you would get thickening. I have no idea. Thanks for that. Good question. Thank you.
Okay, so if that left you intrigued to learn more, we're going to continue along the theme and hear from our additional Breakthrough Prize winner in life sciences, Ulrich Hartl. He's a biochemist and director of the Max Planck Institute for Biochemistry. And along with Arthur, he won the Breakthrough Prize in Life Sciences. But his other work, in addition to the chaperone's role in neurodegenerative disease, also involves really looking at the mechanism behind the aggregation. He's also, in collaboration with Manojit Hartl, has been working and investigating the enzymes in plants that catalyze the production of biomass in atmospheric carbon dioxide. So a very diverse role that he's playing in terms of looking and elucidating some of the science behind our fundamental proteins. The title of his talk is Understanding Protein Folding in the Cell: Opportunities for Improving Human Life in the Decade Ahead. Please join me in welcoming Ulrich.
Thank you very much. Let me also begin by saying how honored I am to receive this award, and it's great to be here together with Art to talk with you. It's especially nice to have this breadth of science from fundamental physics to life sciences and molecular biology at the symposium. what I like to do is to highlight possible benefits of understanding the cellular machinery of protein folding and assembly and quality control in addressing what I believe are two major challenges of human civilization in the years to come. And we already heard about neurodegeneration, the fact that our aging societies are increasingly afflicted by neurodegenerative diseases and dementias. We heard about this from Art just now and from Virginia Lee. So I will try to outline ways in which understanding cellular protein quality control could help us in unraveling the mechanisms that underlie these diseases and perhaps also inform on possible therapeutic strategies. There will clearly be some overlap with what you just heard. And in the second part, I want to address the fact that due to the increasing population on the planet, we know now that agricultural productivity has to increase substantially in order to provide for sufficient food for an ever-growing population. And this means that we will have to improve the performance of photosynthesis, a process in which a particular enzyme that we, and especially my wife's group, managed Hartl's group, is studying, the enzyme Rubisco, plays a key role. And because we now understand the folding and assembly pathway, which is rather complex for this enzyme, we believe that we can now perhaps begin to devise ways to improve the enzymatic performance of this protein. Now, as you just heard, protein folding is a fundamental process in all of our cells. Every organism is the final step in the information flow from gene to functional molecule. Proteins, of course, are the workhorses of our cells. They have to fold into intricate three-dimensional patterns in order to become biologically active. We know now that, contrary to the initial assumption, under cellular conditions, the folding process is not a spontaneous reaction, but it relies for many proteins, probably the majority at some stage of biogenesis, on a pre-existent machinery of molecular chaperones. This does not interfere, obviously, with the idea that the information that specifies the three-dimensional structure resides in the amino acid sequence. Now, chaperones act throughout the biogenesis process of a protein. They begin in at the stage when a polypeptide chain is translated on the ribosome. These are the machines that put the amino acids together to form the protein chain based on the information in the messenger RNA. Then we have HSP70 type chaperones that Art has already mentioned. They will bind to the growing polypeptide chain, binding to hydrophobic sequences, shielding those sequences and preventing the chain from undergoing misfolding and aggregation reactions at a time when the polypeptide is incomplete and does not have sufficient structural information for productive protein folding. Folding can initiate co-translationally for larger proteins that have multiple folding units or domains. We also understand that a subset of proteins need an additional principle of chaperone interaction in order to fold these. This is represented by large cylindrical protein complexes that we call chaperones. These were the types of chaperones that Art and I studied initially and spent many years on understanding in detail. They transiently enclose a partially folded protein in a central cavity and allow it to fold in this space under conditions where aggregation is simply not possible because there's only one single polypeptide chain in that folding cage. So this is a really fascinating principle that nature has evolved to solve the problem of protein aggregation for molecules that have a particularly hard time folding on their own. Now, these chaperone principles have been highly conserved in evolution. They function in ordered and sequential chaperone pathways. to some extent redundant, but sequential steps occur whereby we have chaperones that bind to the nascent polypeptide on the ribosome, and then we have components including the chaperones that act more downstream. Chaperones, however, do not only act when a protein has to first fold upon synthesis. Many proteins have to return to this machinery for surveillance, for conformational surveillance. They require this machinery to maintain their functional structures, especially under a variety of stress conditions such as heat stress, which may induce pre-existent proteins to unfold. Now, a very basic function of these systems is to prevent protein aggregation by the mechanisms that Art has described, by shielding hydrophobic surfaces that would otherwise result or allow protein chains that are not yet folded to clump together, undergoing entanglement. Not only the basic physicists have entanglement, we also have entanglement in this regard. Now, these chaperone machineries, generally, we believe they expand the universe of foldable protein sequences under cellular conditions beyond small proteins that are capable of doing this on their own with high efficiency. The medical significance of these systems arises from the fact that they prevent such aggregation reactions, including, in some systems at least, the formation of the toxic aggregates that are associated with the neurodegenerative diseases that we just heard about. And in order to do so, the chaperones form a much larger network of factors, the so-called proteostasis network, protein homeostasis network, in which there are various functions in de novo folding, in repairing misfolded proteins, and also in disaggregating protein aggregates are integrated with the functions of proteolytic systems, mostly the ubiquitin proteasome system and autophagy, lysosomal degradation, in order to remove in a timely fashion misfolded proteins so that they cannot form toxic aggregates. However, when these systems fail or are overwhelmed, as seems to be increasingly the case in aging individuals, at least we believe that's the case from studies in model organisms, then various types of aggregates can form. As you can see on these slides, they can be soluble oligomeric states, which are believed by most to be perhaps the most toxic forms. And there are end states of aggregates that form highly ordered fibrillar structures. Now, as I just mentioned, we believe from the work of Andrew Dillon, Rick Morimoto, and others that as our cells age, and this holds in particular for post-mitotic tissues, cell types such as nerve cells and muscle cells, which do not undergo renewal by division like a liver cell or an intestinal cell, that as this aging process occurs, the capacity of protein quality control machinery, including chaperones and other systems, undergoes a decline. And this may then facilitate the manifestation of some of the age-dependent neurodegenerative diseases. Now, this is posing a major medical and socioeconomic burden. At the moment, we have in the US approximately 6 million or so Alzheimer's patients, and there will be a doubling by 2050, causing major costs already now, but these costs will reach astronomical amounts in a few decades. Compared to this, by the way, the amount of money that we are spending on research on these diseases or in life sciences in more general terms is a minute fraction. Now, there is not only Alzheimer's disease, there are a multiplicity of forms of neurodegenerative diseases. We have heard about this from Virginia Lee in more detail. But at the core of these diseases is the deposition of aggregates, which have very similar fibrillar structures. And this suggests, I think, that there must be some common mechanisms to many of these diseases underlying their cellular pathology. And it suggests also that we might be finding ways to perhaps treat these diseases in a more general term, especially by utilizing the cell's own defense mechanisms in counteracting this type of aggregation process. various mechanisms of toxicity have been suggested, including interference with the integrity of cellular membranes, the idea that when aggregates form, they may engage in aberrant interactions with key cellular molecules, other proteins, thereby causing multiple interferences with cellular pathways. And this includes also some of the components of the protein quality control machinery itself, as we and others have shown, especially some of the regulators of the major HSP70 pathway, thereby undermining the cellular defense and inducing perhaps a vicious cycle that then results in further and further accumulation of misfolded and aggregated proteins. Now, what can be done? There are various possibilities of utilizing the cellular defense against aggregation. Art has already outlined many of them. We can look for molecules that induce the regulation and the induction of chaperone machinery to augment general chaperone capacity. Various compartments in our cells, the cytosol, the endoplasmic reticulum, mitochondria, have transcriptionally regulated stress response pathways that govern the synthesis of these molecules. So we might be able to find ways to manipulate these pathways so that more chaperone capacity is being produced. Other ways are to manipulate the degradation pathways. Compounds have been developed that activate some aspects of degradation so that misfolded proteins are more rapidly removed. Maybe we can activate disaggregating chaperones. Art mentioned that some of the chaperones can in fact unscramble some of these entangled protein molecules. And also we have been referred to extracellular chaperones that might be particularly relevant in the type of process that Virginia has described, where aggregates are spreading from a particular focus in the brain throughout other brain regions. So the goal would be here to try pharmacologically in various ways to rebalance the protein homeostasis system so that we bring the production of misfolded proteins and chaperone capacity into balance. Now to the second problem, which is seemingly very unrelated, has to do with the fact that we have a growing population. We are presently standing at about 7.7 billion people, 240,000 since yesterday, 70 million so far this year, very close to the population of a whole country of Germany. And this means that we have to substantially increase agricultural production. It has been calculated that we have to increase productivity by about 25% up to the year 2050. However, not only is agricultural production keeping pace with population growth, it is not keeping pace with population growth, but rather is projected over the coming century, over this century, to decline in various regions of the planet due to climate change, particularly in regions of the southern countries, southern hemisphere, some of which are those regions where population growth is in fact the strongest. Interestingly, some regions in the northern part will perhaps even benefit. This has to do with various aspects, one of which might be actually the increase in CO2 concentrations, which of course is the point that I will now be referring to. So what does this have to do with protein folding? The biomass of our planet, all the food that we eat, comes directly or indirectly from the process of photosynthesis, in which solar energy is utilized by plants, algae, photosynthetic bacteria, and converted into chemical energy that drives then a reaction that allows the assimilation of CO2 from the atmosphere, and which is then incorporated into biomolecules, carbohydrates, fats, amino acids, the molecules of life. The light-dependent reaction produces reducing equivalents that go to fuel the light-independent assimilation cycle, and the key enzyme in this reaction is Rubisco. By the way, photosynthesis, of course, also produces as a byproduct the oxygen that we breathe, so it's one of the most fundamental reactions. Now, Rubisco is a complex enzyme. It assimilates 10 to the power of 11 tons of CO2 annually in all these photosynthetic organisms, but apparently that's not enough to prevent the increase in CO2 levels, and this has to do with the fact that Rubisco is actually a remarkably inefficient enzyme. It has a turnover rate of about 3 to 10 CO2 per second, which is a very rather low number for an enzyme. And for that reason, people have been trying for decades to improve the enzymatic performance of this enzyme by engineering methods. But this has been hampered by the fact that we have not been able so far until recently to express the protein recombinantly. That means we could not simply test thousands and thousands of different mutations which would be necessary to find the ones that might improve the performance of this enzyme. This has changed recently, and this is where the knowledge of molecular chaperone systems becomes important, because Rubisco is perhaps the most chaperone-addicted enzyme that we know to date. Not only does this protein require the cylindrical chaperone cage for the folding of its subunits upon synthesis inside chloroplasts of plants, but then it requires downstream up to 4 different types of chaperones, new types of chaperones, that put the subunits of the Rubisco protein together until the final complex forms, which consists of 8 large and 8 small subunits. So now we can take these factors and put them into a bacterial cell, engineer a bacterial cell that makes the whole machinery for the formation of this Rubisco enzyme. This was recent work from Managit's group. And this allows us now to produce Rubisco recombinantly. And I believe this will be the basis for the design of selection screens for mutants of this enzyme. We already have E. coli strains that are dependent on Rubisco activity for growth. There will be a multidisciplinary approach, including perhaps artificial intelligence and other methods to find out what do we have to change in this enzyme to make it a better performer, probably multiple mutations that would be unlikely to happen in the normal evolutionary process because single mutations might already be detrimental. I believe we live in very exciting times where breakthroughs in the life sciences bear fruit to solve some of the most pressing challenges. But unfortunately, our politicians often forget that basic research and funding basic research is an important investment in the future. It is hard to explain to the voters that we just give money to scientists and let them do what they like to do, what they are interested in researching. It is much easier for politicians to come up with all kinds of various programs. But clearly, basic research is the indispensable foundation for the development in pharma and biotech that will eventually advance the quality of human life. Thank you so much.
Your lecture very much, and I had a question. I wasn't certain if chaperones required a sensor of the misfolded state to bind to a misfolded protein, or they somehow can sense the misfolded state and will bind to a misfolded protein.
Most chaperones recognize misfolded states of proteins by interacting with hydrophobic amino acid surfaces that are exposed by the misfolded protein. Generally, this is then also the trigger for the induction of the stress response pathway. Simple models suggest that the transcription factors that regulate these pathways are maintained in an inactive state, being bound to chaperones, and as the concentration of misfolded protein increases, these chaperones are withdrawn from the transcription factor, resulting in its activation and so on. However, we have also observed that some of the most toxic aggregating proteins in disease are not very capable of triggering this response. So we and others are interested in how must we modulate the cellular composition of the protein homeostasis network in allowing it to recognize these dangerous species more efficiently and triggering a stress response. If this were the case, we would probably not have the problem.
Hello. To my knowledge, I'm sorry, I'm over on your right.
Okay.
This is somewhat tangential to the focus of your talk, but one thing you said struck me markedly about the fact that you felt there was a relationship between the fact that neurons and neural cells and muscle cells don't divide, and we see this the degenerative processes in these cells. Well, to my knowledge, we see them much more in the brain, in neural cells, and yet also, I think Joe Altman first showed many years ago that in fact neurons do divide he was using labeled thymidine. It took a long time for others to accept that.
Yes.
But I'm not sure if that's been proven for muscle cells, and yet are there these degenerative disorders of muscles?
Absolutely, there are.
Similar to?
There are, for example, inclusion body myositis, other forms of muscle diseases where you have accumulation of protein aggregates. And in fact, that seems to be oftentimes the source of age-dependent muscle problems that many people, as we age, encounter. Certainly, muscle and neuronal tissues have a much lower regenerative capacity than other cell types, even though some cell division might still occur. So this gives much more opportunity for accumulating damage over longer periods of time. In addition, of course, neuronal cells have an extremely complex architecture, as you, I'm sure, appreciate, whereby transport processes over very long distances play a role. And if you imagine that you have accumulation of protein clumps in these complex cells, it would potentially cause a lot more damage than in a liver cell or in an intestinal cell or a skin cell. Yes.
Lovely talk. So in your Rubisco evolution experiments, one might think that there would be a lot of states that had better catalytic capacity, but would expose too many amino acids and would be degraded. I mean, clearly you can expose those mutants by doing it in proteolytic deficient. But do you think that you could go from a state that's not really acceptable in the wild type? Could you figure out how to re-engineer that high activity state so that it would be more stable?
Yes. So first of all, I mean, the active center of Rubisco is highly conserved in all types of organisms, so we will not be able to make changes in that region, but it's already clear that long-range changes in the enzyme have a strong effect on its affinity for CO2, its affinity for oxygen. I did not mention that not only is it an inactive relatively inefficient enzyme, it is also making mistakes every fourth time or so. It binds oxygen instead of CO2. That's why it's called ribulose bisphosphate carboxylase oxygenase. And that's a wasteful reaction, which people try to engineer away. So far, this has not been possible because you could only test mutations of Rubisco by making a transgenic plant, which is obviously a lot of work. But now I think we can explore a much wider range of mutants and also multiple mutations. We know already that it is actually relatively resistant to mutations that are outside of the active center. Also, you refer to something which I think is very important. We have to understand that proteins and chaperone machines have co-evolved. And we know that the chaperone machinery can buffer mutations in proteins. This has been shown, I believe, the first time most clearly by the work of the late Susan Lindquist, but also in other systems. And so we can explore mutations in the context of perhaps higher levels of chaperone, which would then stabilize folding intermediates or allow certain mutations to reach a folded state that would otherwise be impossible. We can observe, for example, that if you introduce mutations into an otherwise chaperone-independent or relatively independent protein, it creates increasing chaperone dependence that can be observed and can be utilized in order to explore the mutational space of this protein in the context of appropriately modulated chaperone levels.
Here, I'm just wondering if
Yeah.
wondering if the chaperones themselves have evolved to fold spontaneously, or do they depend on other chaperones?
That's an interesting question that we have frequently been asked. The chaperones seem to be no exception with regard to the general requirement of assisting molecules for the folding process. And in fact, there was early on an experiment that Art and I did systematically in order to test the chaperone dependence of the first chaperone that we analyzed in the mitochondria. And it turned out that it required pre-existent chaperone to fold the new subunits of the same molecule that are imported into the mitochondria in this case.
This is a question about diabetes type 2. Islet cell amyloid polypeptide has been associated with development of type 2 diabetes. There's 350 million people in the world with diabetes. Are you studying this protein misfolding problem?
We are not studying this protein in particular, but I know of other groups who are doing so. And thank you for mentioning this. This is certainly another important disease that is associated with protein aggregation. This is not only neurodegenerative diseases, but also obviously type 2 diabetes and another age-dependent disease that seems to be caused, at least to some extent, by the formation of aggregates.
All right, let's thank
Thank you.
Well, thank you. I appreciate the enthusiasm of the questions. It's really fantastic to see the engagement here. And so for our last talk of this first afternoon session, we're going to go back to the physical world. And it's my pleasure to introduce Peter von Nieuwenhuizen, who's Distinguished Professor of Physics at Stony Brook University. He studies quantum field theory with applications in supergravity, supersymmetry, and string theory. And as we heard earlier, he shares this year's special Breakthrough Prize in Fundamental Physics for his work on the theory of supergravity. The title of his talk is The Future of Supergravity, Dream or Reality? It's a very exciting question.
A month ago, I was lecturing at Stony Brook for graduate students, and when I reached the subject of supergravity, I explained how beautiful it is, how interesting. But then one student asked, "Did you write any articles on supergravity?" I was a little bit taken aback, but the students around him pounced on him and said, "Don't you know, he just won the Breakthrough Prize, and he got $1 million for the first article." I wanted to go on, but he asked somewhat incredulously, "Did you really get $1 million for one article?" I told him that I've written about 350 articles, and that therefore I got $3,000 per article. And he seemed to conclude that physics can be a somewhat lucrative business. And then finally, I wanted to go on, but he asked the last question. And the last question was it obvious to write that article? Well, it was not obvious then, but now with hindsight it is obvious. And what I'd like to do now is explain to you the basic principles of supergravity in such a way that if you came here and you didn't understand it, you leave and you hopefully understand it. So what is supergravity? Supergravity is Einstein's theory of gravity, general relativity, GR, with an extra symmetry between the two kinds of particles in our universe, bosons and fermions. I expect you not to understand a word of this. I'm going to explain it. What were or are its aims? On the one hand, theoretical physics, basic theoretical physics, is based on two pillars, quantum mechanics and general relativity. Einstein theory could not unify these two. Our theory is an extension of Einstein theory which marries the two. General relativity, long distances, quantum mechanics, short distances together. The other aim was and is unification of all particles and all forces. Now let me first briefly mention something about unification of particles. Every particle spins around its axis, but the amount of spin is quantized. You have spin 0 particles. These are the Higgs bosons, which have been found. Spin 1/2 particles, electrons, neutrinos. Spin 1 particles, the photon, but also the electromagnetic, the gauge fields for the strong interactions, and the Z and the W bosons, which were found in CERN for the radioactivity. And at spin 2, you have gravitons, which are needed to get the general theory of general relativity of Einstein. But it was always, as Dan Friedman already explained, this gaping hole of spin 3/2. Now, the spin 0, 1/2, and 1 have been unified in something called the Standard Model, which is extremely successful, but it doesn't contain gravity. Supergravity unifies all mass spins from 0 up to 2. But there are also particles which have higher spin, 5, 3, and they have very heavy masses. And string theory unifies all these particles, including the very heavy ones. Now, spin 3/2 is the glue between general relativity and the Standard Model. You may wonder what these asterisks in green are. These are the breakthrough prizes. You can see that string theory in soccer terms leads against supergravity something like 9-1. Maybe that can change. So the contents of my talk are now, I'm going to explain to you what is general relativity, and I think I can do it in such a way that if you didn't understand what it is, you learn what is the basic essence, in my opinion. Then I will tell you about bosons and fermions, but then I will tell you what it means to have a symmetry between particles. And then I will show you two aspects where supergravity does better than Einstein theory of general relativity. I'll show you that quantum corrections are better, and I'll also show you that if you go to higher dimensions, you can unify theories. And I will end with reflections on the state of physics today, my wishes for the future, and also there may be some young people in the audience who want to enter science, so I have some advice for young scientists. So let me explain first what general relativity is. Newton said that two masses attract each other. You see there a planet going around the sun, and the orbits of planets are exact ellipses. This was known since Newton. Einstein said two things. E is mc squared. I'm sure you have heard about it. So energy is mass. And secondly, in 1917 or 1915, he said, energies attract each other. Not masses, but energies. Now, if you have a planet and the sun, there's a gravitational field between the two, and that gravitational field has energy. But Einstein said energy is mass. So between the planet and the sun, there's also mass density. Now, a planet, according to Newton, feels all mass. Not only the mass of the sun, but the planet also feels the mass which sits between the sun and the planet, that mass density due to the gravitational field. And that extra mass leads to an extra force, and that extra force makes the elliptical orbit precess forward, as you can see in that illustration. Now, for Mercury, the effect is exceedingly small. It's 1/50th of the diameter of the sun per century, but the effect has been measured and it completely agrees with Einstein's prediction. So the final message I have is, if you take special relativity, mc squared, E is mc squared, and you take Newton's theory and you reason logically, you get general relativity. Now, what are fermions and bosons? They're the building blocks of our universe. They're named after Enrico Fermi, an Italian physicist, and Bose, an Indian physicist. Supergravity makes a statement that for every building block, there must exist a superpartner, for everyone. So, for example, the fermions we know are quarks, electrons, neutrinos, positrons, which are antiparticle of electrons, and they have partners. But the partners of fermions are bosons, and they have funny names. They're called squarks, S for superpartner, selectrons, and so on. We also have bosons in our world, which we have seen. Photons, quanta of the electromagnetic field, gravitons, quanta of the gravitational field, gluons, quanta of the nuclear force, WZ bosons, quanta of radioactivity. And for those bosons, there are also partners. They are fermions this time, and they have names like photinos, gravitinos, gluinos, and so on. Now, in the first column, all these particles have been observed, and many prizes have been given for them. In the second column, we have all the superpartners. So there's an enormous number of particles to be discovered, but you've heard already the sad news. Up to now, not a single of these superpartners has been discovered. I will address that question. Now, this doubling of particles is not new in the history of physics. In 1928, Paul Dirac claimed that for every particle there's an antiparticle. For a neutrino, there's an antineutrino. For an electron, there's an anti-electron, which is called positron. And sometimes particles are their own antiparticles. Photons are antiparticle photons, and gluons, antiparticle gluons. And gravitons, I could add, antiparticle gravitons. Now, Dirac got antiparticles by taking the square root of the wave operator of Maxwell. So in this single equation I'm going to show, the square root of the Maxwell operator, which is on the left-hand side, is equal to, and on the right-hand side you get single derivatives, ddx, ddy, ddt, ddt, multiplied by objects which Dirac identified as matrices. So square roots give antiparticles. Supergravity claims that if you take the square root of general relativity, you get supergravity. So same idea as Dirac, and also doubling of particles. Now I'm going to explain what a symmetry is. Let me begin with something very simple. If I have a cube and I rotate it 90 degrees, the surface looks the same. You cannot tell whether the cube has been rotated or not. If I take a sphere, I can rotate it around a vertical axis or a horizontal axis, but you cannot tell, if you look at the surface, if it's rotated or not. In the sphere, I can only rotate over finite discrete amounts. In a sphere, I can continuously rotate, but you won't see a difference. So a symmetry means that you cannot tell whether an object is rotated or not. The surface is the same. Now, a local symmetry means that different parts of the sphere you rotate in different amounts, so you deform the sphere. But it is still true that if you look at the surface, you cannot tell if it was locally rotated or not. But local symmetries will lead in general to the word gauge theories, which then Friedman explained, and gauge fields. So the gauge field associated with general relativity are the graviton fields, and gauge fields of local supersymmetry, of supergravity, are gravitinos, as Dan already explained. Now, I'll work this out a little bit. Suppose you see a boson as a hemisphere. For example, I take as hemisphere the graviton, and if I take a vertical axis and I rotate that hemisphere, you won't see a difference of the surface. That symmetry is what Einstein's theory of general relativity is. I could write complicated equations down, but it is really in symmetry, and you rotate something and it doesn't change. But if I take a horizontal axis and I rotate that hemisphere, it looks different. So that's not in symmetry. If I now look at another particle, the gravitino, with a green other hemisphere, Again, if I rotate around a vertical axis, you will not see a difference in the surface, so that is a symmetry. That is the same symmetry as Einstein found in 1915. But if I rotate around a vertical axis, then you will see a difference if it's rotated or not, so that is not a symmetry. So now here comes the simple point of supergravity. If I bring these two particles together, they form a sphere. But a sphere, if I rotate around a vertical axis, you won't see that the surface has changed. That is Einstein's symmetry. But if you now take a horizontal axis and you rotate, you also won't see that the surface of the sphere has changed. And that is the new symmetry for which we got the Breakthrough Prize, supergravity symmetry. Now the change in a boson is a fermion. If I change the sphere, rotate around the vertical axis, the red upper half recedes a little bit and the green upper half comes up. So there's a change what you see in red and green. And the change in the red is proportional to the green times an angle. And similarly, the change in the green is proportional to the red times an angle. But what is new here is the mathematics. The angles over which you rotate and the fermions are described by a new kind of numbers which were invented in the 1800s. And they have the property that A times B is not equal to B times A, but minus B times A. This is essential for these new ideas. Will supergravity turn out to be true? So I'm going to show you two applications where you can see that the theory of supergravity does better than the theory of Einstein gravity. The first has to do with quantum corrections. And all I can tell you is this. The quantum corrections are very complicated, but you can write them in pictures called Friedman diagrams. And these calculations usually give results which are of the form 1 divided by 0. And as probably all of you understand, that is infinity and therefore a terrible thing to happen. So here you see on the left, when I wrote the letter ME, all the calculations or Feynman diagrams you can do if you take the theories of Maxwell and of Einstein, but not yet supergravity, only Maxwell and Einstein, and you do all the loop calculations or the quantum calculations. And indeed, you get this horrible 1 over 0 multiplied by an integer, which is a fractional number, which is a rational number, 137 over 60. So that demonstrates that Einstein's theory is not a good theory to unify quantum gravity with general relativity. But these new pictures, that is what we invented. They have to be added to what Einstein and Maxwell did. And if you add it all up, there's a miracle happening. And when it happened, my heart stopped beating for a moment. They add up to 0. Now 0 for a theorist is the most beautiful number. So this showed that yes, supergravity is a better theory than Einstein gravity in the quantum domain. Now, there have been also 2-loop, 3-loop, 7-loop, and Dan mentioned that already in a particular model. But other models exist where supergravity is not finite. And in string theory, all loops are finite. The verdict is out if supergravity by itself will be all finite, or that one has to go to string theory for all loop finiteness. Let me now show another application of supergravity. Namely, if you go to higher dimensions, you can unify theories. Now let me begin by reminding you that Newton said that what is called the Laplace operator, that is the difficult operator in the first line on the left-hand side, is equal to proportional to the mass m on the right-hand side. That is what Newton said. Now in 1912, there was a Finnish physicist who didn't get much credit. His name was Nordström, and he did a natural extension of Newton's theory. He replaced the Laplace operator by the D'Alembert, so he added that term d2 dt squared in the equation. And that is what Einstein had told physicists in 1905. So Nordström, 1912, 7 years later, just applied what Einstein had told him. It's called special relativity. And instead of the mass, he wrote a relativistic mass, which is Tmm, T mu mu. So that is what Nordström did. But then 1 year later, he had an absolute fabulous idea. He had realized that people could unify space and time into a 4-dimensional representation. So he said, let's look in higher dimensions, 5 dimensions, and look at Maxwell theory in 5 dimensions. It has 5 components on the left-hand side, but then the first 4 components are the Maxwell theory in 4 dimensions, and the last component, he said, that is my field phi for gravity. So he showed in 5 dimensions Maxwell theory gives Maxwell theory in 4 dimensions and Nordström's gravity. But Einstein showed that it was completely wrong because his theory did not predict light deflection at the sun. So it seemed that the whole idea was wrong. But then in 1921, Theodor Kaluza, a mathematician, and Oskar Klein, a physicist, applied the same idea of Nordström, but this time starting with Einstein gravity in 5 dimensions. So in this equation on the left-hand side, you see the 5 by 5 matrix for the gravitational field. And on the right-hand side, you divide that 5 by 5 matrix in a 4 by 4 blocks, g mu of x, that is Einstein gravity, a mu of x, that is Maxwell theory, and then there is a strange scalar field. But notice, that scalar field cannot be gravity because gravity is now g mu of x, Einstein theory. So this field phi of x is in modulus, and we heard from Alex all about moduli, so that is a new interpretation. So this was how higher dimensions, namely 5 dimensions, explain theories in 4 dimensions. Now in supergravity, you can go as high as 11 dimensions. But then you get more complicated theories if you go from 11 dimensions to 4 dimensions. So if you would have gone from 11 dimensions in the early days to 4 dimensions, one would have discovered what is called Yang-Mills theory. It is an extension of Maxwell theory which explains the standard model. Yang-Mills theory is the great discovery. But it was discovered by my colleague at Stony Brook, Professor Frank Yang and Bob Mills, in 1954, having nothing to do with higher dimensions of gravity in a complete different way. So that success cannot be claimed by going to higher dimensions. Now, there are a lot of people working in this field of higher dimensions, and some of these people say the following: the only way to do Kaluza-Klein is through supergravity, and the only way to do supergravity is through Kaluza-Klein. Now, in 1985, a particular supergravity theory, its name is 2B supergravity, in 10 dimensions, was reduced down not to 4 dimensions, but to 5 dimensions. And then a miracle happened. Maldacena, who is not here today, realized that a theory in 5 dimensions can explain, and supergravity theory in 5 dimensions, can explain non-gravity theories in 4 dimensions. This is the ADS-CFT correspondence, which Dan also mentioned. So that was how higher dimensions unify theories. So what directions is physics headed into? You see, we're in a very situation which is even not known by the introduction this morning, when it was said in all fields we have enormous explosion. This is not true in particle physics, experimental physics. We're in a very strange situation. All experiments agree with the standard model of non-gravitational interactions. All experiments have been done agree to many decimals even. And some people say this is a horrible time for physics. It's stagnant. I disagree. It's a golden time for theorists because now you have the freedom to go outside the box of experiment. And indeed, we have a flood of new ideas, of which supergravity is one. Now, obviously, a lot of that new stuff is worthless, and some stuff is very good. So how do we can figure out what is right or wrong? Instead of physical normal experiments, what we now can do is thought experiments, things Einstein initiated. And in particular, the thought experiments we do today is go to higher dimensions and try to see if certain theories are inconsistent. And indeed, most of the new theories can be ruled out this way. Now, I have worked 50 years in this field, and I must say most of my work, and of most people, considers discoveries. We work in a very solid framework of basic principles. Everything is in agreement with everything. All basic laws are very clear. But sometimes there are people who go outside this framework, do something totally unexpected. I can only think of two people. One was Dirac, whom I mentioned, and I could tell you some anecdotes if they're interested afterwards. And the other was Maldacena, who had that ADS-CFT idea. So now we are coming in a new era in physics, where we no longer look at particular models, like the standard model, but we are having a new framework, in my opinion. Supergravity, I would postulate, is a framework. It can be applied in particle physics, it can be applied in cosmology, but I believe it can also be applied in condensed matter physics and even, on the long run, in biology. So no longer particular models, but frameworks and classes of theories. And it may very well be that in the future, breakthrough prizes are for framework of new ideas. So let me now first come to my wish list for the future. The first thing is my dream that one morning I waken up, I take my New York Times from the driveway, and front page news is, a superpartner has been discovered. Now, as you have heard, in CERN, no superpartners have been discovered. In Fermilab in Chicago, none have been discovered. But maybe it may happen in the new accelerator, which is perhaps being built in China. I think we reach the energy range where it can happen. Another thing I would like to mention here is the origin of the cosmological constant. Most people say that nobody knows, and I will tell you, but I will immediately disqualify it, that I believe I know what it is. It is zero-point edges of a supergravity theory at the Planck scale in a curved universe. So what do I mean by that? If you have a flat universe, then you have equal number of bosons and fermions. That's the symmetry between both. Each particle has a zero-point energy. The zero-point energy of a boson is positive and fermion is negative, but equal number of particles and equal absolute values of the zero-point energy, they completely cancel. But in a curved universe, the bosonic zero-point energies get a little bit up, but the fermion zero a little bit less down, and now the sum no longer cancels, but there's a tiny bit left over. So I predict that the cosmological constant is proportional to 1 over the radius of the universe. Now, I have talked with colleagues about that, and they think this is completely ridiculously wrong. And the only argument I have, I can tell you this, is that there's a saying, I'll say it in German and I'll explain it. Sie sind alt genug, sich eine Dummheit leisten zu können. You are old enough to afford a stupidity. And there is some little nice historical story, so again, in the question period, I can say more about it. Dark matter, it would be wonderful if it's gravitinos or superpartners, as Dan already said. So that's a wish list for me too. But even if none of these things will happen, supergravity will remain. First, it solves unsolvable problems in mathematics, and the reason is that supergravity takes also the square root of problems in mathematics which were considered unsolvable. But the square root of these problems can be solved. The chairman of the Breakthrough Committee this year was Edward Witten, and he made an enormous number of discoveries in that direction. And also, a supergravity can just clarify the structure of other theories like the standard model. The reason is that calculations in supergravity are so much easier to compute with. Remember, I showed you that these 1 over 0 terms cancel in supergravity. However, I have to say that Schwinger once told me, and others, that the future lies ahead. And that's, I think, a wise lesson we shouldn't forget. Things always turn out different from the way you expect them to be. So let me close with advice for some aspiring scientists. First, do what you like. Do not do what you don't like. Play, as Dirac said, he likes to play. I would say listen politely to experts, and then forget what he said and go your own way. Second, explain to friends what you don't understand. As a professor, I usually explain to students what I don't understand, and while I explain it, I understand a few more things, and the students understand a lot more, and together we progress. Become an expert in one thing, at least one thing, and then don't let go. In particle physics, there's an enormous competition, and eventually you will be overtaken by a horde of competitors. But if you keep running in the beginning, you have a time that you stay ahead of the crowd, and that's what Dan, Sergio, and I did. And then that's the time you should not let go. You should not take holidays in that time. And when you're overtaken, take all the holidays you like. Learn to calculate. My education was in Europe, where I got a beautiful education, but it was based on ideas and philosophy. And then I came, I've been 50 years almost now in the United States. There the emphasis is on calculations. I think the best is to combine both. But my advice is, if you don't learn to compute, you will never achieve anything in physics. Calculations are crucial. So this is now the end of my talk. And let me quote Goethe. That's why I have given into magic to know what makes the world tick. I could say it in German, but I'll say it in English. Science is a rock of truth in a sea of fake news. Science is culture. It opens windows to see what makes the world tick. Science is the only ideology that is self-correcting. It is needed for society. The future is at stake. Thank you.
In the interest of time, we will take one question before the break, but then we can hopefully talk more about this really inspirational talk.
Well.
Right here.
Who made the statement, Sie sind alt genug?
Ah, yeah, that's a nice term. There were two students in Leiden in the 1920s, and they invented the concept of spin. And they wrote an article about it. Their names were Goudsmit and Uhlenbeck. And their professor was Ehrenfest, who was mentioned in Alex's talk. And he went to the famous Lawrence, who was living a little bit nearby. And the students had sent the article to a journal, but then Ehrenfest showed Lawrence what his students had done, and Lawrence said it's completely wrong. And so the professor went back to the students and said, it's bad news, but your article is completely wrong. And the students said, oh, this is a disaster, because now our careers are ruined. But then Ehrenfest said the famous word, Sie sind jung genug, sich eine Dummheit leisten zu können. You are young enough to stay a stupidity. And I'm old enough. Thank you.
Okay, why don't we thank all of our speakers for this previous session, outstanding work. And I encourage you to grab coffee, talk amongst each other, and be back here at 3:45 sharp, and we'll get started again.
I'm a research scientist and a professor here at UCSF, and also the Vice Dean for Research in the School of Medicine. I have a research lab right across the alley here in Genentech Hall, where we study the molecular mechanisms of cell cycle control. So first thing I want to do is repeat something that Peter Walter said this morning and thank the Breakthrough Prize organizers for putting on this wonderful event. It's so important in this day and age that we focus attention on really basic fundamental science like these prizes do, and I think it's absolutely crucial that the public sees the value of this sort of work. I think the previous speaker said it perfectly in the last session when he said that science is a rock in a sea of fake news, and that's exactly what this symposium means to me, is that we have to resist a little bit this current anti-intellectual tendency that's running through the country. So thank you to the Breakthrough Prize people for putting this together and answering that call. Okay, so why don't we get on with the first speaker, Sergio Ferrara. He's a emeritus professor in the Department of Theoretical Physics at CERN and also at the University of California in Los Angeles. And he is the third out of the 3 physicists who are all given or awarded the special Breakthrough Prize this year for the theoretical discovery of supergravity, which you just heard about in the last talk. As you learned in that talk, the whole concept of supergravity basically extends the standard models of particle physics and into the realm of gravity and introduces this interesting concept that you just heard about the supersymmetry of the 2 particles that are thought to mediate gravity. And I think we'll be hearing a little more about that in this next talk. We have a fun symmetrical title for the talk. I don't know if this is the final title, but here on my little sketch, the title is Supersymmetry, Reflections on the Future of a Symmetry from the Future. Sergio. Thank you.
Okay, I'm very proud to talk today for the Breakthrough Prize, and I'm very proud also that I had the 2 talks by my colleagues and co-laureate of the prize speaking before me. So they let me to give the possibility of giving to you some aspects of the developments of this theory during the years from 76 to now. So my title, my first section of this talk is a 4-string revolution. This is a vision talk, so it's a talk on things which may not occur or may occur. Why first of all, 4-string revolutions? Well, because the supergravity theory was influential and in fact was very important for the 3 major developments which take place in string theory in the last 25-30 years. And these developments in which supergravity played an important role are actually developments which were also recognized by the Breakthrough Foundation with the prize in fundamental physics. The first developments which occurred in 1984, so 8 years after our paper on supergravity was written, was a so-called Green-Schwarz anomaly cancellation with the key forerunner of the GSO projection of Yossi, Scherk, and Olive, which were also 3 influential physicists in developments of string theory. The second development was M-theory unification. You heard already M-theory in the previous 2 talks. M-theory is a supergravity theory which lives in 11 dimensions, so the maximal dimension that the supergravity theory can have. And all supergravity theories in lower dimension, up to 4, can be seen as particular reduction of this 11-dimensional theory. So the Kaluza-Klein program, of which Peter van Nieuwenhuizen talked to you. The M-theory unification then had followed spectacular developments in the so-called IDS conformal field theory correspondence of Joan Maldacena, which was also a Breakthrough Prize winner. And this already was explained by Peter van Nieuwenhuizen, has to do with the fact that the IDS conformal free theory correspondence give a relation of classical gravity in one dimension more of a quantum theory in one dimension lower. I wanted to quote here some physicists who made major contribution in these developments. One, unfortunately, passed away 2 days ago, was Eugene Kremer, which was also my close friend and collaborator, and then Bernard Juliat and Joel Scherk. Well, this is a popular design. In fact, I was surprised that it was not appear in the previous talks, which give a graph on the unification of string theories, or if you want, supergravity theories in a theory which is called M theory. M, like magic or matter, as Witten was usually saying. This theory in 11 dimension has it can be seen as a polygon, and the angle of the polygon are related to different manifestation of this theory. The theoretic string, the type 2a and type 2b string in 10 dimension, and then theories in lower dimension if you compactify some of the dimension of space-time. Then this M theory has an important concept which is used to make this connection of different string theories, which is the definition of duality. Duality has a mathematical definition if you think about Poincare duality, but if you think as a physicist, duality is a symmetry which is manifest in electromagnetism, which has a symmetry in exchanging electric with magnetic charge. So this symmetry, which is the simplest realization, is exchanging of electric and magnetic charge, has actually a more generalization, which can be resumed by the fact that if in a theory you have some parameters, you can have a dual theory where you have that this parameter, instead of being small or large, and there is a mathematical connection between this parameter, which is called duality. And this duality allows you to show that these theories, which are related by the M theory polygon, are actually theories dual to each other in the sense of electromagnetic duality. Well, this supergravity theory, other than unifying in M theory these different theories in 10 dimension and lower dimension, has also a developments by Seiberg and Witten, again, two Breakthrough Prize winner. Edward Witten was actually the chairman of our selection committee, where they use the dualities of the type I was mentioning in theories where you have one or more supersymmetries, which allow you to give exact result in supersymmetric theories. The key ingredient in this exact result is an ingredient which was already noticed by Peter van Nieuwenhuizen in his talk, which is called non-renormalization theorems, namely the fact that in supersymmetric theories, quantum correction are usually milder than theories which have no supersymmetry, and in fact, they are not only milder, but in certain theories, there are no at all quantum correction in certain quantities. And this absence of quantum correction in certain quantities, which you can then compute exactly in the classical limit, are called non-renormalization theorems. Then another developments in which supersymmetry played an important role and has to do with some properties of objects which you have heard a lot during these days, which are black holes. If you have a supersymmetric theories, there are certain particular black holes which usually are not the usual astrophysical black holes, but you have to think that this black hole could have been existed at the beginning of space-time at the Big Bang time. And these are what are called, these black holes are the black holes which have do not have temperature. They have 0 temperature, so they are like, elementary particles. These black holes were shown by counting states in string theory to have a formula which is called Bekenstein-Hawking entropy formula. And this was again a result from 2 Breakthrough Prize winners, Andrew Strominger and Kurup Vafa. I saw them yesterday. I don't know if they're here. And the interesting fact is that there is a mechanism which is called attractor mechanism, which allows you to connect the microscopic counting with the supergravity result, and they agree at least in a certain order of an expansion parameter. In all these events, supersymmetry and supergravity played a crucial role. So you have the IDS conformal field theory correspondence, you have the computation of black holes, and then you have this anomaly cancellation of Green-Schwarz. In all these instances, supersymmetry and supergravity had a crucial role, and this is why I think supergravity has been quoted in the citation of Breakthrough Prize as an influential theory, because it allowed developments. And these developments, in fact, are not only confined to physical theory, but as Peter and Dan already mentioned, they have an important impact in mathematics. The word domain in mathematics received a boost from results in supersymmetric theory, and also classification of supersymmetric theories have to do with the similar classification of groups in the Cartan theory of classical groups. Here, groups are replaced by supergroups, and algebras are replaced by superalgebras. And these mathematical structures are structures which have both commutation and anti-commutation relation, exactly like the numbers that Peter was mentioning. In supersymmetric theories, you have anti-commuting numbers. They are called usually by mathematician Grassmann variable, and these are the appropriate number in which supergroups and superalgebras are used. Now I would like to spend the last part of my talk on some future developments and some present developments which are taking place. One, as Peter was mentioning, is the fact that there is one particular theory, N = H supergravity, which may be in ultraviolet finite. If this is the case, this would be the first example of an ultraviolet theory of gravity, and this would have occurred because in N = H there are much stronger cancellation of quantum divergences than in the case of lower n. Well, this is the multiplet of the particles that you have in supersymmetric theories. These multiplets are labeled by the number of gravitini. You can see that in N = 1 you have 1 gravitino, in N = 2 you have 2 gravitini, and N = 8, which is the maximal possible property, you have 8 gravitini. This N = 8 is closely related to the fact that in a supergravity theory you cannot go beyond 11 dimensions. And so if you compactify 11 dimensions to 4, you get precisely 8 gravitinos. This in some sense is different from standard theory of gravity, non-supersymmetric theory, where the number of dimensions of space-time can be an arbitrary number, can be 100,000 and whatever it is. In supersymmetric theory, the number of dimension is 11, the maximal number. So in some sense, you have only a finite number of theories to investigate. And if you believe the physical relevance of these theories, any theory, including the standard model, should be a compactification of a higher dimensional supergravity. Oh, okay. let me give some thing about the future. Well, the future, I believe, if you want to apply supersymmetry to the real world, the future has to do with also the inclusion in a supersymmetric theory of a model of inflation. This is a very active field of research, especially because of the cosmological data which occur from different sources and which may give prediction on the so-called slow roll parameters in the inflationary model of the universe. Here, I think we have one of the founders of inflationary cosmology, which is Andre Linde, again, one of the Breakthrough Prize recipient. I think the important thing is to see whether there is a standard model for cosmology, exactly like there is a standard model for particle physics, and in particular, a standard model which embraces both cosmological prediction and standard model prediction. The development of the standard model of cosmology was given again by Andre Linde and some other people, one of them, Alan Guth, again, one of the breakthrough recipients. So at the moment, we can make a graph which essentially is similar to the graph that shown by Peter van Nieuwenhuizen, where supersymmetry, if you look at the evolution of our universe and at the forces which are the fundamental forces of non-gravitational interaction, supersymmetry in some sense should explain gravity, eventually gauge group unification for the standard model, and the standard model itself at very low energy, namely the electroweak and strong interaction which are decoupled from gravity as very low energies. Well, I was saying there has been an important developments of theories where you try to embed in a supergravity theory inflationary model for the early universe and particle physics, and they all use the fact that in an inflationary universe you have a Friedman, Lemaitre, Robinson, Walker universe. And then you have to explain different phases where there is always a crucial ingredient, which is the de Sitter or anti-de Sitter space, namely related to cosmological constant. The cosmological constant should be a positive cosmological constant during inflation, and it should was order of magnitude is related to the Planck scale or to the cosmological scale, and should be also another constant, which is the one which is responsible for dark energy, which is the de Sitter vacuum at the end of the inflation, where in fact this de Sitter vacuum in the standard model would be just a measure of breaking of supersymmetry. These two cosmological constants are very different in order of magnitude, almost 10 order of magnitude one bigger than the other, and I think the importance of a cosmological model which include both the standard model and the cosmological model in a unified theory should explain both. And this is something which has been obtained in a particular class of models which are called alpha attractors, and where some of the contributors are Starobinsky, Andre Linde, Renata Kalos, Massimo Porrati, and myself. So let me finish. What I want to say is that if we look supersymmetry for the future, I think supersymmetry for the future should be a theory which encodes the standard model and the cosmological part of the interaction in a unified theory of supersymmetry. So where in fact also the inflaton should come on one of the multiplets of supersymmetry and should have a natural identification in a unique model which encodes the standard model and the cosmological model. So I hope that there will be new exciting developments in this field. Thank you very much.
What's this? Okay. Oh, pass it along to them. Okay. Okay. Back to biology. As you can see, we have a trend developing, physics to biology and back again. We're now going back to biology, although there is a very thin connection between this new talk and the one that we just heard. It all revolves around the Greek word leptos, which is the word for thin or lightweight or small. And in particle physics, a lepton is actually one of the smaller particles, electrons and neutrinos, that we've heard about in the standard model of particle physics. But in this next talk, we're going to refer to the biological term leptin, which is a hormone secreted by fat tissues and other tissues, and that suppresses hunger and is really important in the homeostasis of your body's metabolism. Our next speaker is Jeffrey Friedman, who's a professor at Rockefeller University and an investigator in the Howard Hughes Medical Institute. And he's been awarded this year's Breakthrough Prize in the Life Sciences for his discovery of leptin and for his subsequent studies of the regulation of leptin and its involvement in various genetic diseases of obesity and lipodystrophy. The title of his talk is also symmetrical. It's to eat or not to eat, a key question on the horizon. Okay, thank you.
Well, it's a great honor to be here and be part of such a stimulating and diverse symposium. If you asked the average person on the street why the individual on the left has such a different appearance from the individual on the right, they generally say it's because they eat too much. And that would be right, or correct, but it would ignore the deeper question, which is why they eat too much. And so today I'm going to try to explain what we currently know about the factors that regulate food intake, and then use that as a basis for talking about the prospects for the treatment of obesity. To do that, I'm going to first talk about what we believe causes the problem as an entry point into discussing what we can possibly do about it. Yogi Berra once said that predictions are risky, especially about the future. So I don't want you to consider what I'm about to say as something that is a prediction, rather what I believe to be some opportunities for further research to build on the knowledge we have so far. Now, there are a number of possible explanations for the causes of obesity. They include the notion that obese people lack a level of self-control that lean people have. I find that point of view is more often favored by lean people. It's also said that lifestyle and environment are important contributors, and as well as biological factors. Now, if I were talking about almost any other disorder, people would gravitate to the idea that there's a biological system on which cognitive and environmental factors act. But for reasons that are more sociologic than scientific, people thinking about this disorder tend to consider one possibility to the exclusion of the others. And what I'd like to emphasize today is that there's substantial evidence that biological and genetic factors play a major role in determining how much an individual weighs. Now, there are many ways to assess the genetic contributions to a particular trait. I won't go through all of them other than to say that the most quantitative is to systematically compare monozygotic to dizygous twins. The monozygous twins are more concordant. You can calculate their heritability. And if you do this, what you find is that obesity is the most heritable trait with the exception of height. A higher genetic loading than many other disorders that we commonly accept have a genetic basis: schizophrenia, diabetes, hypertension, alcoholism, and heart disease. Now, to illustrate the power of genes, I want to tell you about a case report that is the story of the young boy whose image I began by showing you. This boy was of normal weight at birth, but he began to develop morbid obesity in infancy. He was already pre-diabetic at the age of 4 when he weighed 90 pounds with 57% body fat. This child was described by Stephen O'Reilly and colleagues in England. And the child came from a highly inbred pedigree, which often indicates a genetic basis. Now, when analyzing the features of this child's phenotype, Stephen realized that the child shared many of the same features with a strange strain of mice that my laboratory had been studying. This is the OB mouse. It's genetically obese as a consequence of a single gene that leads the mouse to weigh 3 times normal and have 5 times as much fat. And in 1994, we identified the defective gene as encoding a hormone that you just heard about, leptin. Leptin is a hormone made by fat that functions as the afferent signal in a negative feedback loop that maintains constancy of adipose tissue mass. It acts on brain centers in the hypothalamus and elsewhere that control appetite. And through this mechanism, body weight can be maintained within a relatively narrow range. I'm going to show you an animation now that sort of gives you a simple view of how the system works. So at each individual stable weight, you eat a certain amount, balance that precisely against the number of calories that are expended, your weight is stable. Now imagine that you lose weight, such as by dieting or starvation, fat mass falls, and leptin falls. A low leptin level is a stimulus to eat more, and this will stimulate one's level of hunger until such time as the fat mass and leptin level return to the starting point. Conversely, if you overeat for a period of time, a period of binge eating, a normal individual will now see an expansion of their fat mass, an elevated level of the hormone. This will too will act on appetite centers to suppress appetite, again returning fat mass to the starting point. So this is a mechanism that maintains fat mass within a relatively narrow range. And this has important evolutionary importance because during our history and that of every other mammalian and vertebrate species, there were dangers associated with either too little or too much fat. Too little fat increases the risk of starvation during a famine, a not uncommon occurrence, but too much fat increases the risk of predation, that you'll be the victim of a predator. And so what this system does is balance those two relative risks, finding an optimal level for a given population in a given environment of fat mass. Now, I don't mean to imply, and I'll emphasize this a bit more in the second hour, no, that this isn't the only factor that regulates appetite, but the genetic evidence says it's an important one. Other factors are involved, but what this system does is essentially resist weight change in either direction, so that if you're going to lose weight by cognitive means, you're going to have to overcome a biological force that's expressed by virtue of this system. Now, knowing this, O'Reilly looked, and I should tell you that the OB mouse that we identified has a mutation in this hormone, so no signal is generated. It's as if the child never generates the signal that there are adequate fat stores, thinks he's starving, and overeats voraciously. And so based on this, O'Reilly went and looked, and it turns out that this child had a mutation in the same gene as was exhibited by the mice. And as I said a moment ago, in the absence of this hormone, no signal is generated that there are adequate fat stores. This is perceived as starvation by brain centers, leading to increased appetite and a massive increase in fat tissue, never making the signal that would shut off the system. In mice, we had shown that restoring the levels of the hormone with recombinant protein normalized their phenotype, so O'Reilly and colleagues attempted the same experiment in this child, and here's the result. These images are the two I began by showing you. This child was leptin deficient and was actually constitutionally lean, as you can see from this photograph, except for the absence of the hormone. Moreover, before the first leptin injection, they gave this child a test meal. At that test meal, he consumed 1,125 calories. That's about half the number of calories an average adult will eat in a day. This 4-year-old or 3-year-old child ate in a single meal and then asked for more food. After a few leptin injections, he now ate an age-appropriate number of calories at the same test meal. Now, I should emphasize that leptin mutations as a cause of obesity are quite rare. That's very typical of hormones, which are poor targets for mutation. So there are only a few dozen leptin-deficient patients in the world, and they all respond similarly to the way this child responds. So while leptin explained, while these experiments or these data, in this case report, tells us that leptin plays a role in human physiology, it doesn't have much explanatory power as a cause for obesity in the general population. So we now ask, what are the levels of leptin in most obese patients? Are they deficient? And as I just alluded to, very few patients, very few patients are leptin deficient. If you now look and compare the amount of fat an individual carries to their leptin level, what you can see is a very high correlation of about 0.9, such that patients with higher levels of body fat have higher levels of the hormone. Now, when you have high levels of a hormone, in the absence of that hormone's effect, which, as I showed you by treating the child, would be to have a lower weight, when you have an absence of the hormone effect and high levels, that generally indicates a hormone resistance syndrome. The classic example of a hormone resistance syndrome is adult onset diabetes. Normally, insulin exerts its action to lower blood glucose, and insulin levels are low. For reasons that are not particularly well understood in type 2 diabetes, there's a block of insulin action that leads to an elevation of blood glucose and a high insulin level. So based on this line of reasoning and the fact that most obese patients have high levels of the hormone, the conclusion has been put forth, and I think substantially validated, that obesity is a result in most of these patients as a consequence of a relative block in its action. Some level of resistance to leptin's effects on the hypothalamus and other brain regions leads to the development of obesity. Now, you might ask then, if a patient already has high levels of the endogenous hormone and is obese, what good would it do to give that patient extra hormone? In the case of diabetes, the answer is not that much. Insulin is not a particularly effective therapy for the 90% of diabetic patients who have type 2 diabetes. So the similar experiment was done here in obese patients a couple of decades ago. In this initial small study, what you can see is that as the dose of leptin increased, there was a statistically significant decrease in weight, but the effect was variable and not especially great, at least on average, because 3 of the patients didn't respond at all. The other 5 there appeared to respond. Lower doses were less effective. So based on these data and the variability of the response, leptin was not developed further as a treatment for obesity. But it raised the question about whether there might be a responder subset. Is it possible that a subset of obese patients might respond to leptin? And further evidence from follow-up trials of this confirmed that, where it was shown, for example, that 12% of patients receiving leptin lost a highly significant amount of weight, more than 10%. 0% of the placebo group lost any weight at all. So the question is, how are we going to? Is there a way to use leptin for the treatment of obesity? And if so, how can we begin to think about its utilization? And the first question was to ask, can we find a biomarker for this responder subset? If we could, of course, distinguish that subgroup in advance, that might provide a means for effectively treating that particular subgroup of patients. I want to return to this slide relating leptin levels to body fat and point out to you that while there's a very strong correlation on average, some patients are quite obese with relatively low leptin levels. This is a logarithmic scale, and you could see between BMI of percent body fat of 40 and 50, which is very high. While most patients have high leptin levels, several patients there have leptin levels below 10 nanograms per mil. This is more or less in the normal range. And I already showed you that at least in the child with absent leptin, leptin is a very effective therapy for weight loss. So the first question was, what if we take the subgroup of patients who are obese but with normal levels, do they respond? And this experiment was recently reported a year ago, and the answer is that it was. This is a prospective study that was done by Amgen and the current holder of the leptin license, Agerion. They pre-selected those obese patients in the lowest decile of leptin, women less than 16 nanograms per mil, men less than 5 nanograms per mil. Women tend to have more adipose tissue and hence higher leptin levels. And what you can see here in this group is that the leptin group in green shows a very significant weight loss of greater than 12%. And by week 32, there isn't any evidence of a plateau. So this would seem to suggest that measuring plasma leptin levels might predict a biological response. I think the evidence is pretty strong statistically and otherwise that it would, as well as based on our current conceptions of the system. So that one possible development as a consequence of the identification of leptin is the opportunity to treat a subset of obese patients. Another possibility that at least has been explored in patients would be to prevent weight gain after weight loss by dieting or other means. I mentioned before that when you lose weight by dieting, leptin levels fall. That's a stimulus to eat more, and that appears to contribute in a significant way to the recidivism for weight regain. Most diets fail within a year or two, and at least the low leptin level appears to contribute to that. So if the major problem is recidivism after weight loss, I think it's supposed to be weight loss. If the issue as it is recidivism after weight loss, could giving and leptin levels are lower after weight loss by dieting, is it possible that giving leptin after an effective regimen of weight loss from a diet might prevent the weight regain? And there's only been one study done of this, and the evidence seems to be encouraging. In this case, there were three groups of patients. They lost their weight, and after the weight loss, leptin therapy was begun. In the blue dashed line, you can see the placebo group starts to regain the weight. And at both doses of leptin, 25 and 100 milligrams twice weekly, you appear to suppress the weight regain. Okay, so those are two opportunities. Now there's a third opportunity that I'll tell you briefly about now, and that is, can we combine leptin with other agents to perhaps restore responsivity to the hormone? And there's been a theoretical basis for considering the possibility of combining leptin with other agents, specifically gut hormones that also regulate food intake. And to explain the rationale for this, I need to tell you that there are two separate interacting systems that regulate food intake. One is a long-term system comprised primarily of leptin and its downstream targets, the purpose of which is to maintain homeostatic control of adipose tissue mass over the long term. The second system are a set of gut signals, hormones, as well as neural factors that report to the brain what the levels of nutrient and recent food intake in the gut are. And it's these factors that regulate meal pattern, how hungry you feel between meals, how full you feel during a meal, so-called satiety. And there's been a reason to think that if you were going to treat obesity, you might need to approach both systems, because after all, if you affect one and lose weight as a consequence, the other might in turn compensate. Now, listed there are some of the gut peptides that are components of this short-term system, leptin appearing to set the gain on that system. I'll mention that one of them, GLP-1, has been shown to reduce food intake, and GLP-1 is about to be approved, or a variant of GLP-1, as an anti-obesity agent. The question here was, if we combine an anti-obesity agent in this category with leptin, would we see an enhanced effect? And I'm going to tell you now about one study that combined leptin and a peptide amylin in humans as a combination therapy that at least tests this premise. And this data, which was published in the PNAS as part of a clinical trial several years ago, now compared the effects of leptin alone, amylin alone, or in green, leptin plus amylin. What you can see is that leptin or amylin alone in this study yielded a little more than 5% weight loss on average. That was better than the previous study I showed you. But what's important here is that if you now combine the two agents, there was a 12.9% weight loss after about 20 weeks. Here again, there appeared to be perhaps a flattening of the response, but it does not yet appear to have plateaued. Now, the fourth possibility would be, if leptin resistance is a result of a block, would it be possible to intervene downstream of the block? That is, if there are downstream pathways that regulate appetite that are distal to the site of the block in leptin action, might that be an effective therapy? Leptin acts on key populations of nerve cells in the hypothalamus. Some of these neurons in green increase appetite, others in red decrease appetite. Leptin works by inhibiting the orexigenic neurons and activating the anorexigenic neurons. And this system has been very well validated by studies that have revealed mutations not only in leptin, but in the leptin receptor, a downstream mediator, MCH, and its receptor, the MC4 receptor, as causing obesity, as Mendelian defects in human that in aggregate account for about 10% of morbid human obesity. So while leptin mutations are rare, mutations in the downstream circuit, the circuit downstream of leptin, are not so rare. And these blocks lead to leptin resistance and obesity. So the question is, could we identify a site that regulates food intake downstream of the block, and might that provide an approach for therapy? Now, much work on the part of our lab and many others now have elaborated a very complex and detailed neural circuit. Each of these circles represents a particular node in the brain in which modulation of specific neurons at that site has been shown to alter food intake. For reasons I won't have time to go into, we identified neurons in one particular locus, the dorsal raphe nucleus, as being downstream, and that's now outlined in red on the diagram there. And we found that in the dorsal raphe nucleus, one population of neurons stimulates feeding and another inhibits feedings, with an architecture not dissimilar from what I just described to you for the arcuate nucleus. And this was well validated. So the question was, could we modulate the activity of these neurons pharmacologically? And if we did, would this be of therapeutic benefit? So our approach has been to try to first test the effect of modulating these neurons using modern methods that can regulate neural activity as a prelude to a drug development strategy. And the better target for these studies appears to be these GABAergic VGAT neurons in the dorsal raphe, shown here in green. And in this study, what we did is took a very well-validated animal model for obesity, diet-induced obese animals that are leptin resistant. They won't respond to extra leptin at all. And in green are the results showing the effect of inhibiting DRN VGAT neurons. So what I just outlined for you is are four potential approaches to use what we've learned about leptin to develop leptin treatments. And I think the evidence in aggregate suggests that obesity has a structure quite similar to that for diabetes. In diabetes, about 10% of patients have what's known as type 1. These patients have absent or low insulin levels, and insulin is a very effective therapy for that subgroup. Type 2 diabetics are insulin resistant. They have high hormone levels, and insulin is a less effective therapy here, and many alternative therapies are now offered for the treatment of that larger subgroup. The evidence for obesity now is that it's similar. We think about 10% of obese patients make too little leptin, either as a result in rare cases of mutations, in other cases low leptin levels as a result of transcriptional defects. And leptin appears to be an effective therapy for that subgroup. For the leptin-resistant patients, who we might consider to be type 2 obese patients, they have high hormone levels, and here we might need to turn to other means, perhaps leptin plus amylin, dorsal raphe modulators, or others. Now, the current recommendation for the treatment of obesity that's been provided to most patients goes something like this: you should eat less and exercise more. This recommendation was first proposed by Hippocrates 2,000 years ago. I'd like to think that modern medicine can do better than repeating a 2,000-year-old recommendation that is no more likely to work now than it was then. And so I'd like to suggest that we think about approaching the treatment of obesity the same way we would any other disorder, and that would include defining the molecular components of the pathway that regulates weight, define the molecular abnormalities in the pathologic state, of course use this framework to study how environmental and cognitive factors might influence, and from this develop rational therapies. So with that, I'll stop, and I'd be happy to take any questions you might have.
Would you just comment on the interaction of diabetes and obesity, in the sense that obesity seems to predispose to type 2 diabetes? Would seem to be some unifying factor there.
Okay, I'm going to put this forth as a hypothesis rather than something that's been settled, but I think there's good evidence, at least, to support it. I wouldn't say it's quite definitive. The current thinking is that fat tissue is actually your friend. Fat tissue represents a safe reservoir for lipid, and that excess lipid in peripheral tissues besides fat is what causes diabetes and metabolic disease. And one line of reasoning suggests that it's only when you exceed the storage capacity of adipose tissue and lipid now starts to accumulate in other tissues that diabetes and other problems develop.
Yes, hi. Thanks for that really clear and good talk. I'm over here. I was wondering if you could comment something that's been in the popular press as well as scientific journals is the role of the microbiome. And I'm sure you know, in sterile mice, you can induce obesity or leanness by those types of things. And how do you think that plays into the leptin system? And is there any hope for that as a treatment for obesity?
So I think there's a very clear role for the microbiome in a number of conditions, immune problems, inflammatory conditions of the bowel. There are studies that suggest that swapping the microbiome in animals can change weight. Some of those studies were done in the OB mice that I referred to that are leptin deficient, and the magnitude of the weight difference depending on which microbiome they have was less than a gram. So I think there may be a role there, but at least based on the evidence I'm aware of, it's not a major role. I think it's certainly statistically significant, but in those same animals where you can vary their weight by a gram, let's say, they'll lose 50 grams if you give them leptin injections. So I think I can't say with certainty that there won't be a role, but the evidence so far doesn't support a major role to my satisfaction.
So it's been shown that epigenetics can play a role in studies of people famished during the Second World War. Even their children have inherited epigenetics that predispose them to obesity. Did you look at that? Is there a relation? Did the epigenetics affect specifically the sites that are correlated with leptin?
So you're talking about the famine winter, and to my knowledge, most of the effects that have been seen were subsequently on diabetes, less so on obesity. Many animal studies have been done by a fellow named David Barker in England, and here again, I think there are certainly effects, but I think quantitatively they're not as great as they are for genetic factors. So I think epigenetics may play a modulating role, but I don't think. My view is that they're not the dominant factor.
You began your talk discussing the heritability of obesity, but you didn't really follow it up by beginning to suggest the genetic factors that actually may modulate the dynamics of leptin action.
That epigenetic factors?
Genetic factors. Polymorphic genetic variability.
Yeah.
Is that a difficult problem because it's so polygenic, or?
So. About 10% of obesity is Mendelian, and it's been very difficult to increase that number as more and more exomes have been sequenced. So the question is, what's going on in the rest of those patients? This is a missing heritability problem that's characteristic of all these disorders, so obesity is not unique in this regard. We speculate about possible explanations. One possibility that it's being polygenic. But one of the things that troubles me a little bit is you see people in the general population with no evident genetic Mendelian defect that are as or more obese than the leptin deficient patients. And so I find it a little unsatisfying to suggest that there's some polygenic combination that's worse than a Mendelian defect in one of the main components. You know, one of the things I was just discussing with Steve O'Reilly, who identified the leptin deficient patient, is what's going on. And we both were wondering actually whether you might in some cases be losing neurons in the neural circuit. There's a lot of precedent for this, including narcolepsy in the hypothalamus, where orexin neurons are missing. This is going to sound like hand waving, but I think that there are ways in which the circuit could be really disrupted based on genetic factors influencing things like the loss of a neural population. We just don't know yet. That should be a soluble problem, or at least an answerable question, but no one has tested it yet.
Okay, time for the grand finale. We're going to get back to one of the New Horizons Breakthrough Prize winners and get back to physics and get up into the sky again for some fabulous astrophysics to finish off the afternoon. Our speaker is going to be Samaya Nisanke, who is an astrophysicist at the Grappa Institute, a center for excellence in gravitation and astroparticle physics at the University of Amsterdam. And she just flew in just for this purpose. I think what we're going to hear today is some really exciting new developments in what's called multi-messenger astronomy, which is essentially astronomy based on different methods of detection than we're accustomed to. I think all of us have looked up into the night sky and looked at the stars, and astronomers for many centuries have been using electromagnetic radiation to detect what's up there and learn about how the universe is structured. But what we're going to hear about today are new methods of not using electromagnetic radiation, but other emissions from distant objects, such as, for example, gravitational waves that we heard about earlier. And also, there are a variety of other interesting emissions that some of those objects are producing and that we're learning to detect so that we can see the sky in different ways. And so, as I said, gravitational waves are the main body of work in this area, and in fact our speaker today was a part of the LIGO team that detected those gravitational waves a couple of years ago and actually won a previous Breakthrough Prize for that collaboration. Okay, so the title of the talk today is New Perspectives into the Universe in the Coming Era of Multi-Messenger Astronomy.
Good afternoon, everyone. I hope you can hear me. So in the spirit of the symposium about the future, I'm absolutely delighted to be here to talk about this rapidly exploding and observationally driven new field of multi-messenger astronomy. And so by multi-messenger, what I'm talking about is radiation that comes from the entire electromagnetic spectrum, as well as gravitational wave spectrum and high-energy particles. Okay, and all the measurements that we're learning from using all these different messengers are allowing us to really make this transformative step forward in having a new perspective in the universe, and in particular in strong field gravity astrophysics. So for the first time, we are actually able to hear through gravitational waves, as well as see through traditional light, the extreme gravity and dynamic universe, as I'll discuss. So 100 years after Einstein's theory of general relativity predicted both gravitational waves and black holes, in 2015, we observed both directly for the first time through the measurement of a pair of black holes merging due to the emission of gravitational waves, essentially taking energy away from the system. And this event occurred 1.3 billion light years away. And the two black holes comprised masses of 29 and 35 times that of the sun. And indeed, the actual duration of the signal lasted only 0.2 seconds that we actually observed. Okay, and within that 0.2 seconds, we actually observed 3 solar masses of energy that were released within this event. So this made this the most luminous event that we've ever observed in the universe. So essentially, the luminosity was about 100 billion trillion times that of the solar luminosity, as well as 50 times as if you sum up all the light from all the galaxies in the visible universe. So I really think, that was absolutely phenomenal. And the event was detected by the laser interferometric gravitational wave observatories located in Louisiana and Washington State. And in gravitational waves, our measurable is this dimensionless gravitational wave strain, which essentially comprises the amplitude and the phase of these ripples in space-time curvature. Okay, so that is essentially what we're measuring. And the gravitational wave strain has a 1 over distance to the source dependence. And essentially, gravitational waves are coherent, and hence the analogy to sound. Okay, so this is why we say that through gravitational waves, we're actually able to listen to the universe. And so far, within the first 2 years of the operation of the LIGO and Virgo detectors, we have announced at least 10 binary black hole mergers. Moreover, on August the 17th, 2017, over 2 years ago, we observed for the first time a pair of neutron stars actually merging 130 million light years away, both in gravitational waves, so you can see the in-spiral in this movie, as well as in electromagnetic radiation in every part of the electromagnetic spectrum. Okay, so as the movie illustrates, the electromagnetic radiation occurred as a result of the fact that there was during this complicated merger, there was essentially gravitationally unbound material that was flung out. And broadly speaking, we observed these two types of matter outflows. One coming from a jet that was moving essentially close to the speed of light, and the second outflow was more spherical, and initially there was this neutron-rich material that then adiabatically expanded and was then powered subsequently by rapid neutron capture nucleosynthesis, and had long been hypothesized that neutron star binary mergers may indeed be one of the astrophysical sites of the heavy elements. And so this event actually represented a multitude of discoveries, so I'm going to focus on in the rest of my talk. And really just to stress that the game-changing sort of aspect of this discovery was actually that we were able, for the first time, to actually pinpoint where the host galaxy and where the event actually happened in the sky by measuring the electromagnetic radiation. And so indeed, the observation of gravitational waves as gamma rays, as well as gamma rays from the signal, initiated this absolutely beautiful and unprecedented campaign of data sets that involved 70 groups over 90 telescopes, both on the ground as well as on space, and over 100 instruments that actually detected the emission in every part of the spectrum. So the optical was detected 11 hours after the event, the infrared at 12 hours, near-infrared at 12 hours, the ultraviolet by the Swift telescope at 15 hours, the X-ray at 9 days, and the radio at 16 days. So this really made this the most studied and a transient event that we've ever observed in the universe. I mean, really just absolutely spectacular. And just to mention that actually this multi-messenger discovery of this event included about over 3,000, apologies because I have a bit of a cold, over 3,000 co-authors actually involved in the multi-messenger discovery, really exemplifying, I think, the global and international efforts of these diverse communities actually coming together and being involved in this discovery. So why are we interested in black holes and neutron stars? Well, black holes represent the extremes of space-time curvature. As we heard today, they essentially have no surface, just an event horizon, which is essentially separating the information from within the black hole's interior with the outside universe. And they're, in some sense, the most simple fundamental objects that we have in the universe. They're entirely described by two numbers, their mass and their spin. Neutron stars, on the other hand, represent both the extremes of space-time as well as dense matter. And so, similar to stellar mass black holes, they actually represent the end states of the lives of massive stars. And their masses essentially range from 1 to a few solar masses, which are crammed into objects with essentially radii of 10 to 15 kilometers, so essentially the size of cities, making their densities as high as if you actually cram the entire of humanity into a sugar cube. Okay, so these really are the most sort of fantastic cosmic laboratories, and indeed the actual mergers of neutron stars make them in some sense, ideal astrophysical colliders. And so, of course, the huge excitement about this field is that from gravitational waves, we're actually able to extract and learn about the source's fundamental properties, such as the black hole masses, the spins, as well as the neutron star equation of state. And from the electromagnetic radiation, we can not only pinpoint where the event happens, but actually say something about the event's energetics, as well as the environment. And so it's really this piecing together of the different pieces of the jigsaw puzzle that allows us to understand compact object mergers within the context of astrophysics, cosmology, and fundamental physics. And so this is really exemplified by what happened in the 2 seconds following the actual merger. So what I'm going to play here in the bottom panel will be that you'll see essentially the time frequency chirp signal, this ever-increasing frequency and amplitude from the gravitational wave merger. And you'll actually hear it. And then 1.7 seconds later, the Fermion integral satellites actually observed a burst of short gamma ray bursts. Now, within those first 2 seconds and these observations, we were able to actually measure and constrain the speed of gravity, or that gravity actually travels at the speed of light, to 1 part in 10 to the 15 for the first time. From the gravitational wave strain itself, we were able to infer the neutron star masses, which are shown by these orange bubbles here, and the blue bubbles up there show the binary black hole mergers that were observed in the first 2 years of the LIGO-Virgo first 2 science runs. So just to express that we really have this diversity in the fundamental properties that we still have yet to understand with black holes and neutron stars. From the gravitational wave strain and essentially triangulating the arrival times of the signals at the 2 LIGO interferometers as well as at the Virgo detector, which just happened to come online 17 days before, we were able to actually pinpoint the host galaxy to NGC 4993 in the Hydra cluster. And this was done at 11 hours by a whole suite of optical telescopes in Chile. And indeed, the continued monitoring of the ultraviolet, optical, and infrared light, as well as looking at the monitoring the spectral evolution of the event over a period of several weeks, really pinpointed the fact that we were looking at a transient event, unlike any that we'd ever seen before in the universe. And so what this movie is showing, and I think this really is just simply spectacular, is actual data from the Very Large Telescope in Chile, the ESO facility. And you can see, I'll start the movie again, but you'll see this initially blue featureless spectrum that is very typical to the photosphere of a sun, essentially rapidly redden over several days' time scale, and you can see these broad line absorption features, which indeed were indicative of the spectral footprints that were expected from neutron star mergers, where this material actually produces the R-process elements, which are shown in yellow here in the periodic table. So these mergers these optical counterparts of these mergers are often referred to as kilonova, macronova, but indeed I actually prefer the term bling nova because, as you can see from the periodic table, these mergers and this particular event really pointed to the fact that we were able to show that we could produce all the heavy elements such as gold, platinum, and silver within these neutron star mergers. So in some sense, the cosmic mines of heavy elements. coming on to the final discovery, I think, or the main discovery of this event. In astronomy, one of sort of the most basic endeavors that we have are to measure the distances to objects. And this is really so that we can actually map out the expansion history of the universe. And indeed, for this binary neutron star merger, we were able to measure from the gravitational wave strain by measuring the distance to the source, having an absolutely calibrated distance, as well as the host galaxy redshift. We were able to actually measure the Hubble's constant from this one event, which essentially encapsulates information about the energy density of the universe. So I think this was really a spectacular, beautiful result, once again emphasizing the beauty of actually joining gravitational wave and electromagnetic radiation. And I'd just like to really stress that all these multiple discoveries associated with this one event have resulted in multiple breakthrough discoveries that are now impacting many different diverse fields, from astrophysics, cosmology, nuclear physics, as well as general relativity and gravitational physics. And really to stress that this is only coming from one event. Okay, so I'd like to end my talk by talking really about the future and where we're going. So since the 1st of April, the LIGO and Virgo detectors switched online again after being offline for 2 years. And in the last 6 months, we have already sent out alerts to the entire world of more than 20 binary black hole merger candidates, as well as more than 5 neutron star binary merger candidates. So we have really entered this new era of population statistics, and in some sense, gravitational waves detections are becoming routine. Now, I think in the next 6 months or so, the next big discovery will indeed potentially be a neutron star black hole merger that we will hopefully observe in gravitational wave and electromagnetic radiation. Why is such a mixed binary so interesting? Well, compared to just smashing 2 neutron stars together, we'll actually be seeing the effect of having the singularity on any tidally disrupted material from the neutron star, and in some sense that it's a cleaner signal in both gravitational waves and electromagnetic radiation. And of course, the unexpected. Critically, we already have several new facilities operating in the optical as well as the radio and the high energy. For instance, the Zwicky Transient Facility, which is located at Mount Palomar. And in the near future, we will have wide-field optical and radio telescopes, such as the Large Synoptic Survey Telescope in Chile, as well as the Square Kilometre Array in Australia and South Africa. And all these instruments are enabling us to have this multicolored movie, as well as multi-wavelength movie of the dynamic universe. So moving away from just having traditional snapshots of the universe. And by having a dynamic movie, this really makes it prime to actually go after these very short-lived transient events and to actually detect more. In terms of gravitational waves, on the ground, in the next 5 to 10 years, we will see the current instruments actually undergoing significant upgrades, as well as new detectors coming online in Japan and India, so the CAGRA and LIGO India projects. And excitingly, in the 2030s, we will see the next, there are two very interesting proposals to build the next generation gravitational wave detectors, the Einstein telescope in Europe, and the Cosmic Explorer, possibly either in the USA or Australia, which will allow us to have a volume reach, which I'll return to, of a factor of 1,000 or greater. In space, in 2034, the European Space Agency and NASA are leading the efforts to launch the Laser Interferometric Space Antenna mission. So essentially here, the aim is to have a constellation of 3 spacecraft separated by 2.5 million kilometers, forming a precision interferometer in order to actually measure gravitational waves, this time in the millihertz regime as opposed to the tens to kilohertz regime that we're seeing currently by the LIGO and Virgo detectors. And so I'll just show the movie again, but sorry. Some nice music, but you can essentially see that you have this triangular constellation that will be trailing the Earth in this heliocentric movie. So in this heliocentric orbit. So really just spectacular in terms of actually measuring supermassive black holes as well as galactic white dwarf binaries. And so coming to really what should we anticipate, and I think for me this is kind of the plot that really, or the schema that really keeps me up at night, as well as I find it tremendously mind-blowing in the 2030s if we do see these ground-based gravitational wave detectors coming online together with LISA. What I'm showing here is essentially in this donut plot, the yellow dots represent binary neutron stars, the white dots represent stellar mass binary black holes such as we've observed, the orange dots supermassive binary black holes of 1 million solar masses. And you can see on the radial coordinate, essentially this is the redshift or the distance, horizon distance away from us. And the blue and the purple and green lines show the horizon distance for the third generation detectors, the Einstein telescope and Cosmic Explorer, compared to LIGO. And the red line shows the horizon distance for LISA. So essentially, within the frequency bands where we're sensitive to the specific masses, we will be observing all the neutron star and binary black hole mergers that exist in the universe. And we should be prepared, and I think this really is sort of the thing that I find just outstanding, to the prospect of measuring 10,000 neutron star mergers per year, as well as 100,000 binary black hole mergers per year from the ground-based third generation gravitational wave detectors. So I really find that, yeah, that really does keep me up at night quite a lot. But of course, if you see, for instance, sorry, because I don't have a pointer, the stars in the middle, essentially the yellow star shows where the binary neutron star that I mentioned, which was discovered in 2017, is located, as well as the white star shows the first binary black hole merger. You can really see the horizon reach. So it's just astounding, but we will not necessarily have the EM facilities to follow up every single event. And so in that spirit, we really are, as a community and as a field, having to think about using these compact object mergers not only as cosmological probes, but also to be able to inform us about something about the large-scale structure and extragalactic astronomy. For instance, how did our own galaxy come to be? So I think this is really where we're facing. We're moving from this discovery era to one of precision physics and being able to answer these fundamental questions. What is the intrinsic nature of black holes and neutron stars? How do black holes and neutron stars drive the evolution of the universe? And of course, using these as test beds to see and measure how fundamental laws of physics interact in strongly field gravity. And just to end with a reminder that we are, as we heard this morning, we are opening this spectrum of gravitational wave astronomy, this several decades in frequency space. I have mainly talked about the high frequency end of the gravitational wave spectrum as well as the millihertz regime, but just to mention that of course there are several other experiments that have already started, such as pulsar timing array that will be observing a much larger supermassive black hole binaries, and that there will be a plethora of other compact object mergers or events, as well as the unexpected that we hope to observe both in gravitational waves and electromagnetic radiation. So just to conclude, I strongly believe the future is both loud and bright, and it's really wonderful, I think, to see all the young people in this room, because this really is a field that we're watching and experiencing a transformative step happening sort of right now, right, this week. And indeed, alerts are happening, I think as I speak. So really, thank you very much.
Question on your right. Just a small point, but regarding discovery for the production of heavy elements in the cosmos, it was interesting that, well, first of all, element 43, technetium, allegedly only occurs, does not occur naturally. It's it's been produced, but element 61, promethium, was not apparently found. Of course, it's a very rare element on Earth. You think that that's a function of the sensitivity of the assay, or is it just something specific about that element that would have not led to its production?
So I should actually mention that within this one event that we observed, only last week was there a paper that actually claimed the detection of strontium in this particular event, but for all the other elements, we have not necessarily observed the individual spectral lines, because essentially when this neutron-rich material is ejected, it's moving at a third of the speed of light. So essentially the lines that you're seeing are Doppler broadened. So it wasn't possible for this actual event to individually identify the elements. So what you're seeing in this periodic table are actually the predictions that have come from detailed nuclear reactions in order to actually, in terms of the predictions that we expected for these kind of kilonova, but they're not actually the measurements from this one event. The only element that has actually been stated as being measured is strontium, but this is, something that, one wants to do in the future with foreseeable events happening.
Over here. Was the frequency of these events shocking to you or the team in general? And do you think that over the course of time, these events are supposed to be more or less frequent as the universe expands and stuff?
In terms of actually observing the mergers?
Or the occurrences themselves, I don't know.
Yeah, so I think if we look at the mean rates, I think in the universe at the moment, there should be a binary black hole merger happening maybe every few minutes, and a binary neutron star about every 20 seconds, if you sort of extrapolate from the mean rates that the 11 events that I've mentioned have actually have actually given. So, I mean, I think it's quite extraordinary. I personally was, because I have been working in this field since I was an undergraduate, basically, I became increasingly more pessimistic as my career progressed as to when gravitational waves would be detected. So I have to say, in some sense, I was really blown away by the first binary black hole merger. I didn't necessarily expect, given the uncertainty in how we actually understand how binary stars evolve, there was 1,000 orders of magnitude uncertainty in the actual merger rates. So even in the most pessimistic scenario, I think, if we hadn't detected anything by the early 2020s, it would have been interesting for constraining binary stellar evolution. But I have to say that the universe is actually quite generous, I think, definitely from this binary neutron star merger. I mean, having worked in this field, I never expected that the first electromagnetic counterpart, the first gravitational wave merger from a binary neutron star would be seen in every part of the electromagnetic spectrum, and that it would be quite so close. I mean, it had a signal-to-noise ratio. I mean, you could see in the chirp of 32. So it was just incredibly bright, and it really was, in some sense, the golden binary. But what we're seeing already in the last 6 months is that there are very interesting candidates, but they're not necessarily quite so close by, and actually finding the electromagnetic counterpart is non-trivial.
Oh, I've got one. Yeah. Okay, that ends our session for this afternoon. It also ends the talks for the day. And so I just want to add my thanks again to the Breakthrough Prize people for putting together just an absolutely joyous and wonderful celebration of basic science, and also mind-blowing. I've had mine blown at least 5 times today. This was really a lot of fun, and let's give the speakers another round of applause. And now we're going to have some closing remarks from the boss. Come on up.
Thanks. And wow, what a cool day. But ain't seen nothing yet. I'd like to especially thank Samaya and all of today's multiple messengers from the future. And just a couple more messages from me. If you're attending the panel discussions, they will begin promptly in this room at 5:30. And a note to our first panelist, Samaya, and the other black hole experts to come up now and get wired for sound. Hopefully the mics won't disappear in the black hole before we start. So we'll see you all very soon. Thank you.